Good afternoon, everyone. Thank you so much for joining, and welcome to the very first ImmunoDiverse Colloquia. My name is Jacqueline Marquez. I'm an SRA in the Fong Lab, and I'm co-chair for the ImmunoDiverse Community Engagement. So we really appreciate all of you for taking time out of your schedule and being here with us today. We really hope that you can stay for the entire event, but if you do have to stay away, we will be posting a recording of this event on our website. So first, I'd like to go over the agenda. We will kick off the event with an introduction and a land acknowledgement. Next, we'll have a presentation by our science keynote speaker, Dr. Brian Bryson, followed by 5 excellent speakers. We will then take a short break and come back to a panel Q&A that includes our 5 trainee speakers. Then later, we'll have a presentation prepared for us by ImmunoDiverse allyship team. We will then announce the community award and end our day with our social keynote speaker, Dr. Dabrowski Herbert, and a group discussion. So now I'm going to hand it over to Dr. Victor Cortez and Elizabeth Mccarthy, who will be giving us an introduction.
Hello, my name is Victor Cortez. I am a postdoc in the lab of Dr. Richard Loxley here at UCSF. I am also a Cancer Research Institute Irvington postdoctoral fellow, as well as a 2021 Public Policy Fellow of the American Association of Immunologists. So we'd like to begin this event by introducing ImmunoDiverse. So what is ImmunoDiverse? We are a collective of trainees and faculty promoting diversity, equity, and inclusion within our immunology community and across UCSF. Our goal is to create an anti-racist and welcoming environment for underrepresented minorities, recognizing that these barriers primarily disenfranchise individuals of Black, Latinx, and Native American descent. And we are committed to tackling racial inequality in immunology. We must acknowledge that the catalysts that spearheaded our ImmunoDiverse program were the social justice movements that arose from the brutal criminalization of Black bodies that has occurred from the very foundation of this country to the present day. These names are just a few of the victims of this racist system. We emphatically state that Black lives matter, and we stand against racism of any form. While police brutality has been a focus of this conversation, we cannot lose sight that systemic racism takes many forms in our society, leading to the marginalization of Black, Indigenous, Latinx, Asian, and other persons. A glaring example of these societal inequalities has been the disproportionate impact of COVID-19 on communities of color. Not only do Indigenous, Black, and Latinx persons have increased rate ratios of COVID-19 infections compared to white persons, but they have a striking increase in hospitalizations, as well as a tragic increase in deaths from COVID-19. And structural racism doesn't just exist out there in society. It's also prevalent within our own scientific research communities. Many of us believe that science is a strict meritocracy, where everyone is equal and judged on their scientific accomplishments. However, these beliefs simply do not match the experiences of many minority scientists. Here, we are showing a few recent articles that detail the many facets of structural racism in science. They touch on topics from the institutional and racist funding gaps that persist for Black PIs to the implicit biases that minority scientists encounter. A common theme emerges from all these articles. Before we can make real change, we must acknowledge the problem, that structural racism exists within science. On March 1st of this year, the NIH explicitly acknowledged this very problem and is pledged to take bold action to dismantle the barriers that impede minority scientists. The NIH currently has an open request for information, and we encourage everyone here to provide their feedback to the NIH. We have just posted the link for this in the group chat. However, the deadline for submission is actually tonight at midnight Eastern Standard Time, so we encourage you to fill that out immediately. Minority scientists also experience the implicit bias of their colleagues, which is particularly damaging and hurtful. We listed just 3 examples from this article of real things minority scientists were told by their colleagues, and which exemplify encounters many minority scientists have had. For example, my colleague told me that Blacks have lower IQ than whites, or my colleague implied that I was hired only due to the lack of diversity in our department, and not because of my scientific accomplishments. And finally, white colleagues have asked why I didn't apply for the minority money. They view these programs as handouts rather than a means to fund exceptional scientists who are too often overlooked because of racism. And finally, one of the most notable challenges is the lack of diversity and representation within academic science. Taking our own university as an example, when we look at Black, Latinx, and Indigenous populations within the US, and then we look at the representation of these groups within UCSF faculty, we see they are strongly underrepresented. We see a similar trend for these groups amongst our UCSF trainees. We know this is not unique to UCSF, and that this lack of diversity is seen at universities across the country. As trainees, it is difficult to exist in the scientific arena when you see so few others who look like you. And it is extremely discouraging to see so little diversity within the faculty ranks. It creates an impression that faculty positions are not for people like you. These are just some of the challenges that motivated us to create ImmunoDiverse, to do the work that will result in real and impactful changes within our UCSF immunology community. I'll now hand it over to Elizabeth Mccarthy, who will tell you more.
Thank you, Victor. My name is Elizabeth McCarthy, and I'm a third-year graduate student in the bioinformatics program. I'm jointly mentored by two members of ImmunoX, Matt Spitzer and Jimmy Yee. And today, I'm going to talk to you about the ImmunoDiverse initiatives that we've developed to address the issue and effects of structural racism in science. The founders of our group wanted to create an action plan that has three key attributes. First, the plan should contain specific, measurable goals so that we can assess our progress or lack thereof towards our goalpost. Second, the plan should be sustainable. What this meant for us was building a community of trainees and faculty leaders who come from both underrepresented communities and also our community of allies. Additionally, we wanted to secure funding sources that would be able to fund multi-year initiatives. Finally, the plan needed to be publicly available. This allows for our work to serve as a blueprint for other institutions, but also ensures accountability by having our group make a public commitment to our initiatives. In this spirit, I wanted to point out our website, which is also going to be linked in the chat, where you can find our plan of action. Before I just delve into the specifics of this plan, I want to introduce our leadership team. ImmunoDiverse was founded by a group of trainee leaders made up of SRAs, graduate students, and postdocs who come from underrepresented communities in science. Their roles are listed on the slide, so I will just call out their names. We have our outreach team on the left, Oscar Aguilar, Priscilla Munoz Sandoval, Victor Cortez, and myself. Next, we have our head of allyship, Joel Babdour, and Jackie Marquez, who alongside with Victor heads our community engagement. We also have Jayla Caston, who's a leader in training. And finally, we have founding members listed here who have since moved on from UCSF to the next step of their career. Brianna Allen, Kamir Hayam Galvez, and Deanna Marquez. We are supported by a number of fantastic faculty and administrators who help turn our ideas into an actionable plan. The faculty team includes Max Kreml, Mark Ansel, Paula Benton-Cure, Sue Lynch, Tiffany Schartschmidt, Jeroen Roos, Vincent Chan, and of course, our wonderful administrator, Isabel Tingen. We work closely with another trainee-led ImmunoX group called IG Equity, which works to promote gender equity within ImmunoX. So our action plan focuses on 5 pillars. The first is representation, and this strives to increase URM representation across the faculty, fellow, and trainee levels within ImmunoX. Next is sponsorship, and this strives to support the success of those URM individuals in our community. Our final areas are outreach, community education, and celebration, and I'm going to dive a little deeper into each of these areas over the next slides. As individuals from underrepresented communities, we understand the transformative potential of higher education, particularly in STEM, and the importance of outreach to recruit and mentor URM trainees into STEM fields. We focused on a cross-generational outreach plan where we partnered with existing outreach programs in order to maximize our impact at the high school level, for community college students, undergraduate students, and also postgraduate individuals. Across all levels, we partnered with the existing programs listed here to fund additional slots for summer or year-long research projects. We work closely with these groups, both to help with outreach and application support to a diverse group of applicants, but also to support programming for the students once they arrive. All of this wouldn't be possible without our amazing outreach team. So this includes the chairs of outreach that I already introduced, but also a group of trainees who are an integral part of this team, including Didi Zhu, Sean Liu, Elma Frias, Miho Tanaka, Maya Arce, Maya Lopez Ichikawa, and Angela To. Our next pillar is community education, which focuses on creating monthly allyship and learning events to share and discuss learning resources related to anti-racism and allyship. In the second session, after the break, our head of allyship learning, Joel Babdour, will lead a session on allyship learning, so you'll hear much more about this initiative then. Our allyship learning team includes many of the leaders who I've already introduced, but also Casey Beppler, Rachel DeBarge, and Molly Bissett. Finally, we're at celebration, which is what we're all engaging in today with our inaugural ImmunoDiverse Colloquia. This colloquia is designed to celebrate the rich scientific contributions of our underrepresented colleagues and also to engage in meaningful discussion on structural racism in biomedical research. We couldn't be here today without our amazing community engagement team. And so I want to give a big thank you to Casey, Victor, Jackie, and Isabel for working so hard over the past year to make this colloquia happen. I also want to thank all of you for coming and participating to make this colloquia a success. Here is really just a sampling of the amazing institutions that you all are coming to be here today virtually, and I look forward to partnering together to advance the mission of ImmunoDiverse. And this brings me to the end and back to our mission statement. ImmunoDiverse is working to create an anti-racist and welcoming environment for underrepresented minorities. We recognize that the current barriers particularly disenfranchise individuals of Black, Latinx, and Native American descent. In this spirit, I want to transition to Dr. Bongiovanni to do a land acknowledgement to acknowledge the traditional custodians of the land that UCSF resides on.
Yeah, Ate. Hi, I'm Tassi Bongiovanni, and that was incredible. And this is really, really inspiring. And so a huge kudos to all of you who organized this and are doing this and a part of it. I think it's amazing. And I'm also really honored to be here. I know that it's sometimes easy to forget the Indigenous component of our lives and of our colleagues. And so I'm really here representing the Native American Health Alliance at UCSF. I'll talk briefly about why a land acknowledgement is important, and then I will offer it. So this hashtag is We Are Still Here. There has been a sometimes actual policy in the federal government in the United States to wipe out Native peoples, either through culture or physically. So this was through residential boarding schools, which were state sanctioned and funded massacres carried out by settlers against Natives. The California missions legalized slavery of California Indians, and there have been many broken treaties across the state and the nation. California itself has the largest Native American population in the country, and there's this sense among many circles that all the real Indians have died off. That's not true. Native culture is alive and thriving in California, from things like Alcatraz and what you may have heard about as the occupation of Alcatraz, from the Klamath Nation, salmon smoking, and all kinds of other ways that Indigenous people are reasserting their sovereign rights. So why do we do land acknowledgements? This is a map of what the Bay Area would look like and the Native people and languages spoken in all of these areas. So you can see here in the Bay Area, we are actually on Ramaytush Ohlone land here in San Francisco. The reason to do these land acknowledgements is to recognize the past and the present, to counter this doctrine of discovery with the true story of the people who are already here. To remind us that colonization happened and is still happening, and to invite all of us to recognize the ways in which we benefit from settler colonialism and to really inspire action against that. It's to offer recognition and respect for elders and for the people who were here before us, and to create public awareness of the history, but also the contemporary Indigenous people who live here. What is a land acknowledgement? So it's offered to open meetings such as this one, ceremonies, lectures, or really any public event. It recognizes the Indigenous people and the importance of land, plants, and animals in this area. The most basic acknowledgements can be very simple, naming the native peoples of the land you occupy, which means you first have to know who the native peoples are of the land you occupy, which many people don't. It can name the common history that is so often ignored, and it can name colonialism's ongoing harms and what that commits us to doing or invites other people to join us in doing. How do you do this respectfully? So you're in relationship with Indigenous people. So this land acknowledgement was created in relationship with the Ramaytush Ohlone elders. So I am not Ramaytush Ohlone, I'm Navajo, but believe so strongly in this that as part of the Native American Health Alliance, we were able to partner with the elders who are Ramaytush to create this. Get involved directly with local tribes efforts. So how are we doing that? We're doing that by helping with the land rematriation from land that the UC systems took from tribes and to rematriate that back to tribes. And to recognize that this is the main goal, is to give land and resources back to tribes and Indigenous peoples. Thank you so much for your time again, and I'm honored to be here. I will now offer the land acknowledgement before the colloquium officially starts. So before we begin, we would like to acknowledge the Ramaytush Ohlone people, who are the traditional custodians of this land. We pay our respects to the Ramaytush Ohlone elders, past, present, and future, who call this place, the land that UCSF sits upon, their home. We are proud to continue their tradition of coming together and growing as a community, and I'll add, with such an important and inspiring mission that you have. We thank the Ramaytush Ohlone community for their stewardship and support, and we look forward to strengthening our ties as we continue our relationship of mutual respect and understanding. And I'll stop there, but again, thank you so much for inviting us as part of NAHA. We're really honored to be here and to be part of this. Thank you.
Thank you, Dr. Bongiovanni, for delivering our land acknowledgement. Next, I am honored to introduce Dr. Brian Bryson while he sets up his slides. Dr. Bryson is the Esther and Harold Edgerton Assistant Professor of Biological Engineering at MIT and a member of the Reagan Institute of MGH, MIT, and Harvard. Dr. Bryson completed his undergraduate studies at MIT in mechanical engineering before obtaining a PhD in biological engineering, also at MIT. After graduate school, Dr. Bryson went to the Harvard T.H. Chan School of Public Health, where he trained with Sarah Fortune and became interested in new approaches to examine host pathogen interactions in TB infection. Dr. Bryson started his lab in the summer of 2018. His lab focuses on bridging quantitative approaches to understand how the immune system eliminates deadly pathogens. I will now welcome Dr. Bryson for our ImmunoDiverse Colloquia Science Keynote. Oh, Dr. Bryson, I think you're muted.
How do I even begin? I have to begin by just thanking you all, thanking the organizers, thanking the people who are here on this call, thanking our mentors, thanking our families, because everybody knows that we don't come here alone. And I have to say also just a deep sense of thank you for this invitation. Like I remember when I got the email, I literally yelled out loud and I emailed my assistant. I was like, clear the calendar. This is the most important thing that I think I'm invited to this year. So I'm just deeply grateful for the invitation because I know how much this matters. And I just know how all of you are excellent and how we need to always continue to push the boundaries of who is welcome in STEM fields. So with that, I'm going to kick off. And so I'm just so excited to be able to present some of the research that's been going on in my lab. where really an inspiring question to us is, what governs the heterogeneous outcomes during infection? And I start with this slide because it's a slide that really inspires a lot of the work in my lab, where we really are interested in how individual phagocytes control the survival of Mycobacterium tuberculosis. And just so you get a sense of what you're looking at in this confocal image, here we took human monocyte-derived macrophages and infected them with a transcriptional reporter of MTB viability. And what I want you to take away is the striking heterogeneity we see in these individual bacteria, where bacteria are all marked by a red fluorescent protein, and those bacteria that retain transcriptional and translational ability are those that express GFP after the addition of anhydrotetracycline. So what I hope you can take away from this slide is that there's an alarming amount of heterogeneity, even at the level of individual interactions between single bacteria and single macrophages. And this is a question that inspires me to this day. And what I hope to show you in this talk is a little bit of how we've started to unpack how complex these initial interactions between phagocytes and mycobacteria are. And I think it goes without saying that tuberculosis is a huge global health problem. It is one of the leading causes of infectious disease death. It's estimated that nearly a quarter of the global population is infected by Mycobacterium tuberculosis. This leads to about 4,000 deaths per day. And one of the really striking things about TB at the human population level is not everybody infected with TB actually goes on to develop active disease, which is characterized by symptoms such as weight loss, coughing, and fatigue. Now, not all hope is lost because we do have effective antibiotic treatments for drug-sensitive TB, but this drug treatment regimen is actually quite demanding. It's about 6 to 9 months of a 4 antibiotic regimen. And most importantly, and I think what would be transformative for all of us, is a protective vaccine for tuberculosis, which we don't yet have. And today I want to just kind of acknowledge that many of these questions about how we're going to get to vaccination really relies on our understanding of how the immune response handles Mycobacterium tuberculosis. So that's what we don't understand, but I just want to make sure we're all on the same page about what we do understand about this immunological life cycle. So TB is transmitted via cough. So somebody infected with a bacteria will cough. This aerosolizes the bacteria into droplets. Those droplets then make their way into the lungs of another bystander individual. The bacteria in these droplets make their way into the alveolar space of the lung, where this alveolar space is patrolled by one of my favorite cell types, the macrophage. And this macrophage will then engulf the bacteria. And this engulfment or phagocytosis results in the initiation of an innate immune response. And this innate response really drives the formation of one of the hallmarks of TB disease, which is this granuloma, which is really just a collection of macrophages. But what's really amazing about TB is that this granuloma goes on to evolve. And when it evolves, we actually see the recruitment of other lymphocytes to the site of infection. So we can have what's called this lymphocyte cuff of B cells and T cells. Now, one of the amazing things about a granuloma is that a granuloma can go on and take various outcomes, both from a microbiological standpoint and from a biophysical standpoint. So for example, a granuloma can then go on to become fibrotic, or it can become necrotic. A granuloma can fully sterilize infection, or a granuloma cannot fully sterilize infection. And all of these types of granulomas can be occurring in the same individual. So there's a striking heterogeneity at the population level, but even within a given individual, this heterogeneity exists. So this is what we understand about this life cycle, but there's a lot of unknowns. And anybody looking for a TB postdoc, hit me up. But because I think there's a lot of unknowns that we can begin to think about. What are the host determinants of control? How is bacterial virulence regulated? And what are those properties that really enable the bacterium to survive? And many of these knowledge gaps are fueled by several limitations, not limited to the fact that TB is by no means a model pathogen. We have to study it in a biosafety level 3 environment. It takes, it doubles every 22 hours. And so some of these challenges just create, in my world, many new opportunities for us to begin to try to address these questions. So one of the questions that we started off my lab addressing in my lab was really this simple question: is what does the early immune response to MTB look like? And I want to acknowledge that a lot of previous efforts have been applied in this space, but I think new measurement techniques have really begun to really ask us to reevaluate how we set up our TB infection models to begin to even explore what this might look like. So I just want to give us a kind of a recap of what we understand about TB transmission dynamics these days. And so one of the things I want everybody to know is that TB disease in the non-human primate can best recapitulate some of the clinical spectrum that we see in human individuals. So this is work done by Joanne Flynn and Sarah Fortune, where in the non-human primate model of TB, they use genetically barcoded strains of TB that they bronchoscopically instilled into the lung. And what's amazing about that is that even very few, less than 10 CFU, can cause fulminant TB. But really interesting, because these individual bacteria were genetically barcoded, they could use PET-CT to look at where the individual granulomas were, and then ask at the time of necropsy about how many bacteria founded an individual granuloma by deep sequencing. And really the important thing to take away here is that most granulomas are founded by a single bacterium. So this means if we kind of zoom back in time, what it really means is that an individual granuloma is founded by the interaction between a single bacterium and a single macrophage. Okay, so this suggests that these experiments suggest that yes, we can drive TB disease with very few bacteria, but what's the evidence that actually an infected individual actually expels very few bacteria? And for that, we'll go to South Africa next, where in South Africa at the University of Cape Town, this really amazing piece of kind of, I call it an engineering marvel, was constructed, where individuals who, upon their TB diagnosis, were actually asked to enroll in a short-term study. And this short-term study involves the following. They sit in what's called this cough box, which is a room. You can see this chair here in 7, and this TV for the at number 5. And what they're asked to do is they're actually asked to sit and watch TV for 2 hours. And what this box is doing in the process is collecting the expelled air. And really by using novel kind of enzymatic probes to look at bacterial enzymatic activity specific for MTB enzymes, they can take this air that's been expelled, condense it down, and then ask, with using these probes, how many bacteria does an infected individual expel in those 2 hours? Now, what kind of blew my mind is what the quantitation looked like at the end. And just to give you a punchline, here in the span of 2 hours, active TB patients, they were able to detect bacteria on the order of tens of bacteria. So this again really kind of sets up this idea that really the initiating kind of infection is probably driven by very, very few bacteria. Okay, so this is kind of the problem that we were confronted with. And my lab is a biological engineering lab, and what we really like to be able to do is build models that can, in the laboratory, that can help us understand what might be happening at these early stages of infection. And so that again was the question that we set out to answer. And we were really inspired by this conceptual model from Ron Germain's lab, where what happened in this set of experiments led by Rachel Gottschalk were that they took murine bone marrow derived macrophages and took KLA, a TLR4 ligand. And what they did was something very simple but elegant, is they did intracellular cytokine staining for either CCL3 or TNF with a dose response of KLA. And what I hope that you can take away from this graph is that as they increase the concentration, really here at 100 nanomolar, they see nearly all of the cells are responding. But as you begin to titrate down this concentration of KLA, you see this really impressive, almost switch-like response, where even like at 0.1 nanomolar, only about 18% of the cells are responding by producing these two cytokines. So we're really inspired by this conceptual model because it really sets up this question about what is the quality and quantity of signal that's being driven in to, and how does that translate into an immune response thereafter. Okay, but the real challenge here is that unlike toll-like receptor 4, MTB is thought to signal via toll-like receptor 2 and other pattern recognition receptors on the surface. And what I want to do here is I just really want to acknowledge the superhero of the story who did all this work, Sydney Solomon, who's a 4th-year graduate student in my group, where Sydney was really excited about translating this conceptual model into thinking about how this might be applied to Mycobacterium tuberculosis. And so the first experiment that Sydney did was she took human CD14 monocyte-derived macrophages. So these are macrophages that we're isolating by CD14 positive selection, differentiating with MCSF for 6 days, and then asking, okay, what might happen when we look at, instead of TLR4 responses, look at TLR2 responses. And in this case, what Sydney used as a TLR2 ligand was PAM3CSK4. And so what you're looking at here are flow cytometry plots. Here we'll just start with the unstimulated cells. So in the absence of TLR stimulation, you can see that most of the cells are not producing any TNF alpha. So our x-axis is TNF, our y-axis is just side scatter. Now, as we increase this concentration of PAM3-Cis at 0.1 nanograms per mil, we're not really seeing very much. But really impressively, when we go to higher concentrations, again, we see this thresholded and switch-like response. So at a nanogram per mil, about 4% of the cells are responding, and at 100 nanograms per mil, and this is really important, about 52% of the cells were responding. So what this says is, yes, we're seeing a conceptually similar response to toll-like receptor 4 signaling, but one of the really interesting things that we observed is that even at really, really high concentrations, even if we go to a microgram per ml, one of the things that we found is that even when we go to these really high concentrations, in the toll-like receptor 2 response, we actually were not seeing, and at really high concentrations, all of the cells responding. Again, suggesting that there's this kind of inherent heterogeneity in the toll-like receptor 2 response. So just to summarize what we've seen thus far, we do see this switch-like response, but really at high concentrations, not even all the cells have the capacity to respond by producing TNF alpha. Now, I won't show you the data, but we actually confirm this across a whole variety of toll-like receptor 2 ligands. So this is not exclusively limited to PAM3CSK4. This is actually a very general property of toll-like receptor 2 signaling. Okay, so we see that there's this switch-like threshold, and one of the arguments is that this switch-like threshold is the pathogenic concentration. But what I showed you earlier is that TB can cause disease with a single bacterium. So the question is how does this conceptual model now translate to intact bacteria? And here, what we're going to use is fluorescent MTB, because one of the challenges with infecting at a low multiplicity of infection, as you would want to if you're trying to model these early responses, is that a very small population of cells is actually going to be infected. So to overcome that challenge, what we use is we use fluorescent bacteria that allows us to distinguish between directly infected cells, those that are GFP positive here on the right, and bystander cells that we'll call GFP negative. And then we can quantify in these two different populations how much TNF alpha is being produced. So to jump to the punchline here, if we look in human monocyte-derived macrophages about 24 hours after infection, what you can see here on the x-axis is GFP. So we see, again, these two populations of uninfected bystanders and directly infected cells. Now, if we look on the y-axis, which is TNF here, what you can see is really striking. Is that about 1 in every 50 cells that's infected with MTB actually respond by producing TNF alpha. Now, if we increase the MOI to about even an MOI of 10, we do see an increase in the percentage of cells that are responding, but it's not very dramatic. Okay, so this was really impressive. We're really excited to see this result. But the question is here we're using a commonly used lab strain H37RV. And a question that we next wanted to ask is whether or not this was just a response that we're going to observe in this lab strain, or if this was more generalizable to distinct clinical strains. So what we did is we were fortunate to obtain clinical isolates from distinct TB geographies, so to speak, and we repeated the same experiment. And what I just want you to take away is that if we look at these 3 different clinical isolates, what you can see is, again, in this GFP positive population, very few of the cells are actually responding by producing TNF alpha. So this is a common theme across these TB strains, that TB seems to not be able to elicit a really robust TNF alpha response. Okay, but here's a problem. So the problem is there's actually 2 ways to interpret this data. So a question is this low TNF production a consequence of MTB-mediated evasion? And so you can imagine there's actually 2 models here. One is that TB is a weak agonist, and number 2 is TB is evasive. And so we needed a set of experiments to try to be able to discriminate between these 2 models. And why is this an attractive model in the first place is if we actually go to the Legionella pneumophila literature, it's well characterized that Legionella actually has secreted effectors that can disrupt host protein translation in cysts. And what you can see here is if we actually just label, and this is not we, this is work from Sunny Shin's lab, Ralph Isberg's lab. If you actually take fluorescent Legionella, infect, and then look for new protein translation using these novel probes of protein synthesis, what you can see is that those cells that are actually truly infected with Legionella actually have no new protein translation. And so this was a really exciting conceptual model for us, because not only could you look at individual cells by microscopy, you can extend this to flow cytometry. And so a question for us then became, could we extend this conceptual model to explore whether TB may be doing something similar? And so to make a long story short, that's what we did. So we used these kind of novel protein translation probes, and I'll just walk you through the data here. So if we look at a sham infection in this dark gray, we see that we get this kind of level about 10 to the 4 for new protein translation. If we use a known protein translation inhibitor, cyclohexamide, we see a decrease as one would expect. Now, if we look at either the bystander cells or the directly infected cells, there's two things I want you to take away. There's no statistically significant difference between the DMSO treated sham cells or infected cells, and there's no difference between the infected cells and the bystanders. Again, suggesting that TB is not disrupting global protein synthesis. All right, but that doesn't necessarily get us in the clear, because it's possible that TB may have specific ways in which it can disrupt TNF alpha production specifically. So a question was, how are we going to figure that out? You could imagine doing ribosome profiling, but that's a lot of work. And so Sydney came up with this really clever experiment that I love. And Sydney's idea was that we could take GFP MTB, infect at a low MOI, where we know we're going to get these two different populations, bystanders and infected cells. And then we can kind of come in with a positive control, where our positive control that we know we should see TNF alpha production is if we add PAM3CSK4 and then add secretion inhibitors and ask the question of, is there a difference between the bystander cells and the directly infected cells in their ability to produce TNF alpha? And so a hypothesis here is that in these bystander cells, there's no TB present, so they should be able to induce the production of TNF alpha. Now, if TB is evasive, what you would expect is that when you add PAM3CSK4, you won't see an increase in TNF alpha production. Now, if TB is instead a weak agonist of TNF alpha production, you would expect when you add something that you know should drive TNF alpha, you'll see this increase. So that's the experiment Sydney set out to do. So again, here on our y-axis is TNF, our x-axis is MTB GFP. So you can again see here that in the absence of any stimulation, you see very few cells producing TNF alpha. Now you add PAM3-CIS, you see this increase. Again, here, when you infect with TB and don't add any additional ligands, you see that very few of the cells are actually producing TNF alpha. Now, really strikingly, when Sydney went on to add either PAM3-CIS at a nanogram per mil or 100 nanograms per mil, what we found was really striking. And what you can see here is that when we actually add PAM3CSK4, we see that the GFP infected cells have absolutely no defect in producing TNF alpha. They can do it no problem, suggesting that it's not that TB is evading this process, it's just naturally weak at agonizing this. Okay, so this is awesome. We can show that TB is very weak at inducing TNF alpha production. And I like to be super transparent about our data. And so the thing that I'll tell you here is that we really wanted to look at other cytokines as well. ICS in human monocyte-derived macrophages in our hands has been really tricky. So we've not found amazing clones to be able to look at other cytokines as well. We have some data with IL-8 and we're excited to look at other cytokines, but I just want to be clear that we've only been able to really explore this in deep depth for TNF alpha. Okay, so as we think about this input-output relationship between adding MTB and looking for TNF alpha, I'd be remiss if I didn't acknowledge that going from infection with TB to the production of TNF alpha is really mediated by this very complex immune response. So even if we just think about toll-like receptor 2 signaling, which is not the only receptor that's going to be driving the response to MTB, we can think about all what's happening downstream at the transcriptional level to drive the production of TNF alpha is really mediated as early as the formation of the mitosome, which will drive the activation of transcription factors such as NF-kappa B and the phosphorylation of some of these MAP kinases such as TNF alpha, or MAP kinases such as ERK and P38. Okay, so a question is could we actually begin to discern where might there be bottlenecks or defects in kind of the natural kind of relay between these pattern recognition receptors and these downstream transcriptional responses. So what we've seen thus far is that TNF alpha is not well induced, but the question is what happens further upstream? So what we sought to do is we sought to examine activation of either NF-kappa B, vis-a-vis looking at I-kappa B alpha degradation, phosphorylation of ERK, and phosphorylation of P38. And for the sake of time, I'll just show you a few snapshots of the data and get to the punchline. So here, if we use soluble PAM3CSK4, we see exactly what we would hope to see. So we see in this purple curve, we see this degradation of I-kappa B alpha, which is transient, which allows NF-kappa B to translocate into the nucleus and drive transcription. If we look at either phospho-ERK or phospho-P38, we see this characteristic kind of, early peak in phosphorylation, which is attenuated over time. So this suggests that our assay works. And so now we wanted to be able to extend this with our GFP-MTB to look specifically in those cells that were infected with MTB. And so I'll just jump to the punchline here. So we looked at all these additional nodes. And what I want to focus on is phospho-P38. So again, I'm showing you PAM3CSK4 here. And you can see, again, this really robust phosphorylation of P38 by PAM3-Cis. But really strikingly, when we did this experiment and looked at GFP-MTB, what you find is that there's basically no phosphorylation of P38 at early time points, which again raises a really important question because, how we leverage these templates of toll-like receptor signaling, we really need to begin to evaluate how individual pathogens may vary. So, and I just want to again emphasize here that when we looked at I-kappa B alpha degradation and ERK phosphorylation, TB did those two just fine. Okay, so again, the question becomes, is TB inhibiting this, or is TB just a weak agonist of this process? And so to begin to explore those questions, we used a very similar experiment to those that I described earlier, where we can take TB-infected macrophages and then stimulate with something that we know should drive P38 phosphorylation. And in that case, what we did is we used anisomycin, which is a known P38 agonist. So here on the left, here are just sham infected cells. And you can see when we add anisomycin, just for even just 30 minutes, we see a really robust increase in P38 phosphorylation. And when we look at either the bystander cells or the directly infected cells, we see that these cells actually, when you add anisomycin, the cells retain their ability to drive P38 phosphorylation. So what this suggests really is that if TB is inhibiting P38 phosphorylation, it can be easily overcome. Okay, so I set up this idea that really we're using these in vitro models to begin to explore the immunological life cycle of MTB. And the question you might say is, okay, Brian, this is all in a dish. Can you give me any evidence in vivo that this is something that we would expect or something that's consistent with observations in humans? And so that's what I'd like to do next. And so actually, if we look in the non-human primate, it's actually really striking how consistent some of our observations are with an intact organism, the intact response in a non-human primate following low-dose CFU infection. And so what was done in this experiment is that there were serial blood draws over time. You can see the time post-infection here on the x-axis. And what was done at each of those time points is they asked, how many differentially expressed genes can you detect? And really what I want you to take away from this is that it takes about 10 days before you see a really robust transcriptional response in the periphery. Okay, Brian, but TB's in the lung. So I think this data is really suggestive, but it'd be even more powerful if we had data directly from the lung to corroborate this kind of hypothesis. And so we can go again, return to South Africa, where what was done is they took humans that had been previously vaccinated with BCG, and they asked them to enroll in a human lung BCG challenge. And so the time point for collection here for both proteomics and RNA sequencing is 72 hours post-infection. And they initially started with a dose of 100 CFU. And their argument is that, okay, we used 100 colony forming units and we saw about 3 differentially expressed genes after 72 hours. And so their argument was, let's go higher. So they went to 1,000 and they still didn't see very much. And so then they went to 10,000, and that's really when they began to see a robust transcriptional response and protein response in the lung, which again is consistent with an idea that actually TB or mycobacterium more broadly may not be that potent at inducing many really robust early either transcriptional or protein response following infection. So what I hope to have shown you today is that, that we have these inflammatory thresholds that really shape not only the toll-like receptor 4 response, but also toll-like receptor 2. My thinking right now is that MTB immune evasion really starts with being in this subdominant regime. Our transmission is probably in a really kind of sub-threshold regime. And, it's going to take some time until TB can replicate enough to really cross that threshold. If we think about a pathogen that doubles every 24 hours, that we really have this variability that we need to think about overcoming in the pathways that drive TNF production following TLR2 stimulation. And who knows about the other variability in other networks. And importantly, that while we can use these templates for toll-like receptor signaling that have been developed by really exquisite research, both at UCSF and elsewhere, that we really need to understand how pathogen infection may vary. And I hope the P38 result shows you that some of these responses can be very distinct. And lastly, that there's this in vivo evidence for muted responses to mycobacteria. So I don't like to close talks by just telling you, okay, this is what I hope to have shown you. I also like to give you a flavor of where we're heading next. And so for those of you who are MAP kinase aficionados, one of the things you'll notice is that I really focused on ERK and P38 in my talk. And that's because at the beginning, the phospho-junk antibody we had was literally junk. So we weren't able to see really robust signal, but we've tested a bunch of antibodies. So we're really excited about now being able to go back and do these experiments and ask, does junk look like the ERK response or does it look like the P38 response, and really be able to build out a better understanding of kind of the MAP kinase arm of the response to TB. And then secondly, really dig deep about what are the transcriptional programs or just programs writ large that are controlled by P38. And this is in part inspired by previous work that really demonstrates that in other organisms that loss of P38 is really important for pathogen control. And then, as I mentioned earlier, one of the limitations of these experiments is that we really could go take a really deep dive, given the tools we had access to look at the response of TNF alpha at the protein level in these cells. And so one of the things that I'll be excited about to share in about 6 months, is that we've now come up with these new single cell technologies to be able to really transform how we look at host pathogen interactions by single cell RNA sequencing technologies, by really being able to overcome one of the key challenges in host pathogen interactions, especially with TB, which is trying to understand for a pathogen like TB, which is really difficult to lyse and will never lyse in your 10x lysis buffer, how we can convert this GFP signal, so to speak, into something that we can detect by transcriptional profiling without having to do flow sorting. So with that, I want to close, but I always have to acknowledge the trainees who do the work. So like I said, this work was pioneered by a really amazing graduate student, Sydney Solomon, who did literally every experiment that I described to you today. I just want to acknowledge my entire lab. The reason I do this job is because my lab is an amazing place, and it's because I have an amazing group of trainees. Being a junior faculty, being a trainee, being anybody in these times are hard. So I'm deeply thankful for my extended scientific family. I'm thankful for my funding sources, and I'm thankful to you all. And I just want to say, as I close, again, a deep thank you. This is a really meaningful invitation, and I, more so than anything, really look forward to all of your success in the future. So with that, I'll close, and I'm happy to take any questions that you have.
What a fantastic talk. Thank you so much, Dr. Bryson. So Casey Bepler is going to be moderating the Q&A session, but I'll go ahead and start it off with a question. So I was really intrigued by your data on the sort of sigmoidal response of TNF to the TLR2 agonist. And it kind of made me think of, like with enzyme kinetics, when you have cooperativity. So I wanted to ask what you think the mechanism is between that sort of switch-like action. Do you think it's something about the receptor and some sort of cooperativity, or is it getting signals from other cells? I was really intrigued by that, not even in the context of TB, to see that sort of behavior.
Yeah, so Elizabeth, that's an amazing question. And I'll start with saying that that I really think that it's really happening at this MAP kinase level, because in fact, there's a lot of literature about ultrasensitivity and kinetic dynamics of in enzyme biochemistry, and really driven by the activity of relative activity of the kinases and the phosphatases, especially in these really complex MAP kinase pathways. And so that's one hypothesis that we're really excited about, and has been previously demonstrated that as you alter MAP kinase activity, you can really switch where that threshold lies. And actually, if you look, for example, in raw macrophages, which have aberrant MAP kinase signaling relative to like murine bone marrow derived macrophages that you derive from the femur, you see this really striking shift in where that threshold lies. And if you start to modulate MAP kinase signaling, there's a really clear contribution to where that is placed. So one model that we're really excited about is really digging deep into these MAP kinases more deeply. But you can imagine that there are other places where in this network where there could be other drivers of kind of this sigmoidal response, but MAP kinases is where we're starting.
Thanks so much, and I'll pass it off to Casey for the rest of the questions.
Thanks, Elizabeth. My name is Casey Bepler. I use she, her pronouns. I'm a grad student in Max Krummel's lab at UCSF. I am an organizer on the allyship and colloquia team. Really excited to be doing questions today. Thank you so much, Dr. Bryson. So first question we have is, what was the control group for the TB room model? So that cool setup you had at the beginning. And could the same model be used to measure other pathogens like virus?
Yeah, so the control group were confirmed PCR negative individuals who had no microbiological evidence of TB infection. So those are the control groups. And, you know, can it be used for other detection of other pathogens? I certainly think that it's possible. I think that one of the reasons that this technique worked super well for MTB was the generation of these probes by actually some of your West Coast colleagues, Carolyn Bertozzi, who actually developed these biochemical probes that can look at specific mycobacterial enzymes exclusively. And so I think that if you had for, you know, certain pathogens, a really sensitive detection agent that was really specific for that pathogen, you could imagine how it would generalize. But that's really the key. That's the key hurdle, technical hurdle to overcome, is having a probe that's sufficiently specific for your pathogen of interest.
Great. Next question. Did you ever try to see if infecting with dead MTB can cause the TNF alpha induction? For example, inactivate the pathogen by UV or fixing with formaldehyde?
Yeah, so that's a really good question. Thank you for that one. So we've done a similar experiment. So we've done the experiment two ways. So if you heat kill, which obviously is going to make a lot of structural changes to the bacterium, you do see a more robust TNF alpha response. So that certainly happens. Now, if you, by contrast, per formaldehyde fix, the trends are not different. So we again see a very muted response, because our hypothesis is, okay, yes, we can do this anisomycin experiment to show that it can be overcome, but is there like some low level thing that MTB is doing that like could be disrupted by fixing the bacterium? And the short answer is that we didn't see a real difference in the nodes that we queried when it came to looking at the PFA fixed bacteria.
Interesting. Next question. Does the amount of receptor TLR2 change as a function of inflammation? Would a positive feedback of increasing TLR2 explain the switch-like behavior?
Yeah, so that's a good question. So what I can tell you is that we looked at TLR2 expression levels prior to stimulation, and it was, you know, like all the cells were TLR2 positive, and they were all TLR2 positive, and we looked a little bit at, you know, post stimulation what the levels look like. And they do change and at the surface. But I think the really interesting thing about toll-like receptor 2 is it has like a surface lifestyle and it has an endosomal lifestyle. So I'd be remiss if I didn't like go back and redo those experiments by looking for total TLR2, because you could imagine compartmental signaling is going to matter a lot. But yeah, so that's one of the things that we're really excited about really understanding more deeply, is about where is TLR2 signaling? Is it signaling from the surface or from an endosome? And what are the relative consequences of where it's signaling from? But right now, what I can say is that the levels do change at the surface following infection. In fact, when you infect with TB, the levels go up a little bit more. So yeah, we're still sorting through all of that. I mean, pattern recognition receptors in TB is a very interesting question for us because, you know, when we think about an intact pathogen in and of itself, like it's not only toll-like receptor 2, there's all the other PRRs that could be engaged. So yeah, so we're really excited about exploring these questions more deeply. And thank you for the question.
Awesome. Well, we have time for probably just one more question. So the next question is, how do you think the heterogeneity of granuloma type, i.e. sterile, fibrotic, productive, in MTB infection is related to the properties / microbiology of the individual infecting bacterium and the macrophage response? Are bacteria that get sterilized better at, oh, same kind of thing.
Yeah, so I really like a conceptual model where we can think about hitting the lottery in terms of what macrophage you end up in, how well poised that macrophage is to mount an early immune response, and really who it recruits in to help form that granuloma. So while I kind of presented this model of like macrophages get recruited to form the initial granuloma, like some of our other data where we see, for example, that one of the really things that macrophages, TB infected macrophages do really well is they secrete CCL7 and IL-8. So monocyte chemoattractant and neutrophil chemoattractant. So one of the concepts that I'm really excited about exploring more deeply is really trying to understand like what the secreted cytokine milieu for an individual macrophage does for kind of recruitment. But I think that there are so many layers of heterogeneity. I think bacteria are naturally heterogeneous, mycobacteria specifically, because they grow and divide asymmetrically. I think all of the single cell RNA sequencing data that everybody is doing in almost every lung pathology suggests that there's heterogeneity in that myeloid compartment. It's just not just like this alveolar macrophage monolith. So you can imagine there are so many dimensions. And so it kind of like begins to grow exponentially in terms of the potential combinations that you have. So I definitely think that that is contributing, but how we study that is going to be the real hard challenge.
Yeah, that sounds fascinating. I'm sure getting into some microscopy and the spatial aspects of it all is going to be really interesting as well. Awesome. Thank you so much. We actually had a ton of questions for you. I'm sorry to the audience that we can't get through more of them, but at this time we have to move on. So thank you so much for that great talk, Dr. Bryson. I'm going to ask Jackie to come back on as we transition to our trainee speakers.
Thanks, Casey. Okay, so I'm excited to present our first speaker, Gail. Gail is a fifth-year PhD student in the BMS program working in the laboratory of Dr. Tiffany Scharschmidt. Gail is also a National Science Foundation Graduate Fellow. She graduated from Fresno State University, and her path to UCSF was facilitated by her participation in the Summer Research Training Program. Gail is passionate about community outreach and has done so to students at Cal State and the Bayside Academy in San Mateo. Please join us in welcoming Gail as she tells us about her work on CD4 T-cell responses to commensal bacteria during colitis.
Hi, I just wanted to make sure if my video is okay or my presentation.
Yeah, we see it.
Okay, perfect. Just want to make sure I'm on the right one. Okay. Hi, everyone. Thank you for the kind introduction, Jackie, and I'm glad you all could make it. I'm Gail, a fifth-year grad student in Tiffany Scharschmidt's lab, and today I'll be talking to you about some highlights from my thesis project. So during my PhD, I really became interested in how the immune system maintains good relationships with their tissue-resident neighbors, the commensal microbes. Our lab in particular is interested in what separates bacterial friends from foes in the skin. So we know that from previous work that early life is crucial to establishing these relationships, and maintenance of these relationships in the form of immune tolerance comes with development of commensal-specific Tregs that persist later in life. These commensal-specific Tregs are involved in suppressing potential inflammation brought on by effector T cells and neutrophils. So this is sort of a bit of an oversimplification, but for now, we'll focus on these particular cells for the rest of the talk. So now, how exactly do we study these antigen-specific responses to commensal bacteria? We largely rely on sort of in vivo models to dissect these responses. And then in our lab, we utilize a defined bacterial antigen in the form of a common skin commensal, Staphylococcus epidermidis, or Staph epi as I'll call it from now on, that has been engineered to express a model antigen, 2W. Then we identify these antigen-specific CD4 T cells through the use of MHC class II tetramers, which allows us to really find these rare cells, sort of finding a needle in the haystack. So during her postdoc, our PI Tiffany elegantly demonstrated that early life was really key to establishing immune tolerance to the skin commensal Staph epi. To show this, she started with two groups, one group of wild-type Black6 pups that were pre-colonized with Staph epi in early life, and another group that did not get pre-colonized. She then lightly tape stripped and colonized both groups as adults, what we call our Staph epi re-challenge phase, from now I'll call this, and compared the skin immune response to Staph epi. Pre-colonization resulted in a robust antigen-specific Treg response that coincided with a reduction in inflammation, one of the measures being reduction in neutrophils in this model. So our other projects in the lab have largely focused on the mechanism behind this establishment of tolerance to Staph epi in early life. However, we were also particularly interested in the mechanisms by which this established tolerance could be either disrupted or lost in adulthood. In particular, we had an interest in the crosstalk between the gut and the skin. We know from the literature that during homeostatic settings, these tissues are largely compartmentalized, meaning the resident immune cells interact with their resident local microbes. However, we wondered whether in disease settings that this compartmentalization could be lost, especially during inflammation. So clinically relevant to us was the knowledge that some subsets of IBD patients could present skin manifestations which only show up in adulthood. And our hypothesis then was sort of that the underlying intestinal inflammation could really lead to skin inflammation as a pathogenic response to what's normally a commensal microbe on the skin. Oops, sorry, I went all the way back. So we chose a DSS-induced colitis model to loosely model IBD and test this hypothesis. So what we did was we first pre-colonized two sets of wild-type Black6 pups with Staph epi and in adulthood induced colitis by giving DSS via drinking water in one of the groups during the Staph epi re-challenge phase. I should mention that DSS is a chemical that sort of destroys the intestinal barrier and induces inflammation. It's a very commonly used model in the field of colitis. And so colitis, which is in orange here compared to blue, really altered the quality of the immune response to Staph epi on the skin, as shown here by reduced percentages of commensal specific Tregs in both the skin draining lymph node and in the skin. Comparatively, colitis did not reduce the polyclonal Treg percentage in either of these tissues, which really means that this is an antigen-specific response. Colitis also resulted in increased neutrophilic infiltration and IL-17 alpha production by polyclonal CD4 T cells in the skin. And this really, we wondered why this was happening and whether any sort of systemic inflammation was sort of enough to induce this response. So really, although inflammation stems from the gut in this model, this also brings into circulation an increase in pro-inflammatory cytokines and inflammatory cells such as monocytes and neutrophils that could be doing this response. So then we next tested whether or not a model of systemic inflammation could alter the response to Staph epi. And in this case, we used the LPS-induced model of sepsis. We again pre-colonized two groups of mice with Staph epi and then re-challenged in adulthood with one group receiving LPS injections in pink. And then we were able to induce inflammation in these mice in data not shown here, but really neither the quality of the antigen-specific response or the recruitment of neutrophils into the skin as a response to Staph epi re-challenge was altered in these mice. And this really suggested to us that there was more to colitis that was responsible for the disruption of the established tolerance to skin commensal. So how does colitis exactly alter this response? So although largely a skin commensal, we found that Staph epi was sort of present in the intestines of our mice, and this then resulted in altered local gut responses. Staph epi has been found to thrive in sort of weird conditions, such as in the infant gut, where there's not a lot of competition. It's also been shown to be sort of prevalent in times where barrier is disrupted in the gut. And as we show in our model, we can really see Staph epi thriving in the gut of these mice. But not only is it present in the gut in much larger quantities in mice with colitis, as shown here in the stool CFUs, but it also induces an augmented antigen-specific response in these same mice. And we also see that the quality of the antigen-specific response is altered locally as a response to this gut colonizing Staph epi, as shown here by the reduction of Staph epi-specific Tregs that we also saw in the skin and skin-draining lymph nodes. Because of the expansion of both polyclonal and Staph epi-specific T effectors in the gut, we wondered whether CD4 T cells could potentially be trafficking from the gut to the skin. And so in simple adoptive transfer experiments, we took CD4 T cells from the gut draining lymph nodes of donor mice that either received control or DSS drinking water during the Staph epi re-challenge phase. We then transferred these cells into congenitally labeled recipients that then got re-challenged with Staph epi, and then we harvested skin draining lymph nodes and spleen to compare these tissues. I note here that we also took the spleen as sort of a comparator tissue, because normally in these adoptive transfer experiments, you may not get exactly the same amount that were injected or that persist in the mouse. And so if we look at the percentage of, or the proportion of the congenitally labeled donor cells that persist in the skin draining lymph node over the spleen, we find that CD4 T cells from DSS treated donor mice actually showed greater propensity to traffic to the skin draining lymph nodes compared to control donor CD4s, suggesting that colitis could be altering the homing potential of these CD4 T cells. I should mention here that we also have some interesting preliminary findings from adoptive transfer of antigen-specific CD4s using the OT2 model, where we saw similar results, although I'm not showing that here. Finally, we wanted to know what colitis-driven signals could be tuning the response to altered response to Staph epi. And so we really found that IL-1 beta is increased in the serum of our mice with colitis, which is not surprising because this coincides with increased levels in the gut mucosa of patients with IBD. We also know that IL-1 beta plays a role in Th17 induction, so that was interesting to us. And that, as we previously found in our lab, it drives responses to pathogenic skin bacteria during early life. And so, interestingly, there's also been literature on the role of IL-1 signaling on CD4 T cells directly, which prompted us to explore this pathway. So in deciding this, we considered the tools that were already available to us and the fact that CD4 T cells are not involved in development of the acute model of DSS colitis that we've been using. So then we wanted to test whether IL-1 signaling directly to CD4 T cells played a role in this differential response to Staph epi during colitis. And to do this, we bred CD4 Cre mice to IL-1 receptor flox mice. We then compared Cre positive and Cre negative mice that were pre-colonized in early life and either re-challenged during DSS colitis or just received controlled drinking water as a baseline comparison. We found that there was no baseline differences in response to Staph epi in the Cre negative and Cre positive mice, but that there was a reversal of our commensal specific Treg phenotype during colitis. In data not shown here, we found that polyclonal Tregs remained unaltered, so this was really, again, an antigen-specific difference. And interestingly, the reversal was due to the reduction in numbers of Staph epi-specific T effectors. So finally, I'm just showing here our working model, showing that upon DSS colitis, we get this impaired intestinal barrier that allows for disruption of the normal gut environment, including the expansion of Staph epi, which is normally skin commensal. This then results in recruitment or induction of inflammatory immune cell subsets, such as T effectors, into the gut. And these CD4 T cells could have an increased propensity to traffic from gut to skin, and along with altered cytokines such as IL-1 beta, really promote differential commensal specific responses in a distal tissue such as the skin. So hopefully I've shown here some of the brief potential mechanisms behind the so-called gut-skin axis that occurs during colitis. and most importantly, the antigen-specific responses that occur. We have some other experiments in progress that I won't have time to go over here, but please feel free to ask any questions. Finally, I'd like to acknowledge all the people that have helped me with this project. Thank you, and I couldn't have done it without your support. I am ready for questions.
Great, thank you so much, Gail. I'll actually start us off with a question. So this special relationship, it seems like, between the gut and the skin is really interesting. So I was wondering, do the CD4 T cells from the DSS treated mice increase at other sites or just the skin? And if just the skin, or if they especially go to the skin, what directs them there?
Yeah, so we haven't looked at other tissues. We're thinking of looking at the lung as well as a potential reservoir for these cells. And we think that the additional, so I should mention, when we re-challenge these mice, we actually tape strip the back of the skin, the back skin. So we actually provide an extra signal there. And I've done experiments where I compared tape stripped and non-tape stripped mice that both got colitis. And this was only, this reversal or this reduction in the Treg response was really in the tape stripped mice. So it could be recruitment of other inflammatory cells into the skin that then sort of provide a positive feedback loop to where they recruit T effectors as well.
Right. Interesting. Thanks. So a question from the audience. Everyone can feel free to send questions via chat to Question Master. But our first question is, did you look at chemokine receptors on the transferred cells?
Yeah, so we did start looking at those. We started sort of comparing CCR4 versus, which is mostly a skin-directed chemokine, versus alpha-4 beta-7, which is a gut-directed chemokine receptor. And unfortunately, our CCR4 staining was not great. So if there's anyone here who has had good experience with CCR4, I would love to hear from you. But we're also trying CCR10 next, which is another potential receptor.
Great. Since Gail is a co-host, you should be able to direct chat Gail if you would like to give her suggestions if anyone has them. So that's available. Probably our last question here is this specific for Tregs? Do other T helper populations show similar traits such as CD8 T cells?
Right, so we haven't really seen a difference in CD8 T cells at all. So this really seems to be, at least in the polyclonal level, because our tetramers are only detecting the CD4 T cells.
Great. Thank you so much, Gail, for that great talk. I'm going to transition us on over to our next trainee speaker, who's going to be introduced again by Jackie. Okay.
Thanks, Casey. So the next speaker is Dr. Rogelio Hernandez Lopez. Dr. Hernandez Lopez is a postdoctoral fellow in the laboratory of Endothelium. He has been doing pioneering work investigating CAR T cell therapy, some of which was recently published in Science. Early in his postdoctoral training, Dr. Hernandez Lopez was awarded the UC Mex's postdoctoral fellowship. He is also a Cancer Research Institute Irvington postdoctoral fellow, and he was a finalist for the Burroughs Wellcome Fund's Career Awards at the Scientific Interface. He has also been awarded the American Society for Cell Biology Public Engagement Award. And if that's not enough, Dr. Hernandez Lopez has also been involved in promoting STEM education to the community through the Clubes de Ciencia México initiative. Please join us in welcoming Dr. Hernandez Lopez as he shares his groundbreaking work on CAR T cells with improved antigen density discrimination capacity.
Hi, thank you so much for the introduction. And I just want to make sure that I'm showing the correct.
That looks good. We can
okay, see. Perfect. Yeah. I am really excited to tell you about my work. And today I'm gonna mention how we engineer T cells to be able to discriminate cancer from normal tissue based on the amounts of proteins that are expressed on the surface of T cells. So let me begin just by mentioning this new field that has made remarkable success in cancer therapy, which is the use of synthetic biology and in particular, these chimeric antigen receptors, which you can then redirect the cytotoxic activity of a T cell towards cancer. Like these receptors identify particular proteins on the surface of cancer cells, and then you can identify and target cells. And in particular, this has been remarkable for the treatment of lymphoma and leukemia. But if you try to use the same approach against antigens that are expressed in solid tumors, then there is multiple challenges. And one of them, and the focus of my work over the past few years, is that there is very, very few tumor-specific antigens. And what that means is that like the antigens that you would like to target, for example, HER2, EGFR, or some of the ones that I show you here, they are not only expressed on tumors, but they are also expressed on normal tissue. And one difference, though, is that the levels of the expression of these proteins are different in tumors versus normal. So most of these antigens are overexpressed in tumors, but they are also expressed at low levels in normal tissue. And the problem is that if you try to engineer now T cells to recognize one of these antigens, then things are complicated because they don't have the ability, these CAR T cells, they don't have the ability to discriminate based on this antigen density expression. And as you can imagine, this could be problematic because that has resulted in problems of on-target but off-tumor toxicity that in some cases has been lethal. So the question I asked when I started my postdoc was, can we control the T cell activity to discriminate the targets based on antigen density? And in particular, what you would like to do is to reshape this behavior of the engineered T cell from a linear response to ultrasensitive response. I'm really glad that we heard before about ultrasensitivity and these sigmoidal responses, because that is one feature that we wanted to engineer in order to achieve this discrimination. So I'm gonna mention now how we actually achieve this ultrasensitive response. And as you could imagine, we borrow concepts such as cooperativity. And one of the ideas that we had was to build a circuit. And in order to build this circuit, we may use a new class of receptors, the so-called synthetic notch receptors that were developed in the lab of Wendell Lim a few years ago by Leonardo Morsut and Paul Roybal, now a professor here at UCSF. And the idea of these synthetic notch receptors is that you can convert a binding event into a transcription activation event. With these synthetic notch receptors, you can then modulate the expression of any payload of your choice. And in this particular case, we decided to control the expression of a CAR. And both the synthetic notch and the CAR are targeted towards the same antigen, with the additional twist that the synthetic notch receptor is of a low affinity, while the CAR receptor is of high affinity. And the design or the idea behind the circuit is going to be shown in the following schematics. If the synthetic notch receptor encounters target cells that have low levels of the antigen, because the recognition domain is of a low affinity, one would expect that the frequency of that interaction is low, and therefore there shouldn't be CAR expression. But if the same synthetic notch receptor encounters now target cells that express high levels of the antigen, then you should expect this robust induction of the CAR. And because the CAR is of a high affinity, that should lead then to this positive feedback and the activation of killing and proliferation and the cytotoxic response of these T cells. So in order to test this idea, first we built a series of engineered cell lines. And in this case, I use as a model system the antigen HER2. And the difference between these cell lines is 100-fold difference in expression. And these lines that express the high levels match the levels that you find in several cancers such as breast or ovarian, for example. And the lines that express the low levels also match the levels that you find in several human tissues. At the same time, we also built a series of recognition domains based on the very well characterized Herceptin antibody against HER2. And these recognition domains also expand a 100-fold difference in affinity. And so we could culture T cells with these engineered target cells and use flow cytometry, and found that indeed you can now reshape the activity of these T cells to discriminate high levels of the antigen versus low levels. And I don't have time to show you all the details of these experiments, but in the recent publication, we showed that by tuning the affinity of either the CNotch or the CAR, you can modulate the behavior of this circuit cascade. And this is in remarked contrast of what you will find if you only use constitutive expression of a CAR, because regardless of the affinity, they don't have any density discrimination. In another set of experiments, we perform microscopy assay and cancer cell lines that express either high levels of HER2 or low levels of HER2. And earlier I showed you that constitutive CAR expression doesn't have any discrimination, but the circuit strategy that I mentioned earlier, where you use a synthetic notch receptor of low affinity inducing the expression of a CAR, only kill the high HER2 cells. So in the last set of experiments that we carry out with several tumor models, we implanted these high HER2 tumors on one flank of animals and low HER2 tumors in the contralateral flank. And this is in NSG animals. And several days after we inject the CD8 and CD4 T cells engineered with this circuit, and we found that the only tumor that is killed is the high density HER2 tumor, whereas the low HER2 tumor continues growing as when you inject these animals with untransduced T cells. And these results are consistent with finding a larger number of T cells, and also those T cells have higher expressions of a CAR only in the high density tumors. And in contrast, in control experiments, if you inject animals with a low affinity CAR, both tumors get killed. So today I told you that using synthetic biology and a very simple concept such as this circuit strategy, one can reshape the activity of these T cells to now have ultrasensitivity and being able to discriminate normal tissue from tumors. And that is one of the main problems of these therapies. But of course, there is multiple others that I'm excited now to continue working towards leading these therapies to more solid tumors. So in the last minute, I would like to invite you to know a little more about this organization that is very close to my heart. And I think this is very relevant for the event today. I think when I was a grad student, along with friends and colleagues, we established and started an outreach program. And the idea here is very simple, is how we can invite more high school and college students to pursue careers in STEM, professional opportunities in STEM. And the model is very simple. Every year we invite graduate students and postdocs that are doing research here in the US or in Mexico to design these intensive 1 week long workshops that are delivered in multiple cities across Mexico. And I invite you to, if you are interested in this opportunity or learning how you can get involved, please talk to me. I will be happy to chat about it. This model now has been replicated in 8 countries in Latin America and Ibero-America. So I'm very excited to continue expanding these opportunities. With that, I'd like to thank Wendell Lim, my advisor, my collaborators, Seb Gardner and Carles Axila, and all my collaborators with the recent publication, and of course, the funding, the Cancer Research Institute and the UC Nexus Fellowship. I'm happy to take any question. Thank you.
Great, thanks, Rogelio. So we have some questions here. First question, so there's a lot of data that points to a problem of CAR T cells in solid tumors becoming exhausted by low-level signaling or even tonic signaling in some cases. Have you looked in those tumor experiments at any indicators of exhaustion in your own system?
Yeah, that's a very good question. Thank you. I have been starting to look into that, and something that I can tell you at the moment is that actually the strategy of the circuit of not having the CAR all the time express on the surface of this T cell already is an improvement on all these markers of exhaustion. And there has been a couple of recent publications. I think they are going to come out at the end of this month from Cole's lab, Cole Roybal and the Lim lab, pointing towards this, not that the strategy of gating the CAR expression is already has an advantage for that. And what I'm trying to do is to get a better understanding on how these density levels affect those markers. But yeah, that is a very good question.
Great. So our next question, I'll read it as is. Beautiful talk. Can you also engineer CAR T cells to preferentially home to specific tissue sites, i.e. to the tumor endothelium?
Yeah, that is a very good question as well. And what I can tell is that the lab of Wendell Lim and others are very interested in using technologies like this to actually do tissue-specific selectivity and homing or induction of different payloads. I think using synthetic biology and knowing what is specific about these different tissues and using perhaps logic gates, or different strategies that allow you to only target specific tissues, then you could probably direct these T cells towards those tissues. In addition to that strategy of the recognition, I guess communicating these T cells, engineered T cells with endogenous T cells, to attract endogenous immune cells could be also a very powerful strategy to do this type of a specific targeting.
All right, thank you. Next question and comment. This is a nice system. Are you concerned that if these CAR T cells are only activated in the presence of high levels of antigen, it will be easier for the tumor to escape by slightly downregulating the level of antigen?
That is also a very good question. And I think something that I've been exploring lately is exactly that part of what is the affinity of the CAR that is optimal, or the half-life of the CAR that is optimal to deal with heterogeneous levels of density expression. At this point, I only show the extreme cases, being able to the very, very high and the very, very low. But as you point out, one would like to also be able to target intermediate levels, and make sure that even if the tumors downregulate, they don't escape. And perhaps this strategy of targeting a single antigen could be helpful, but also you could imagine using additional antigens to avoid that type of escape.
Great. So we've unfortunately reached the end of our time for questions. I want to thank everyone for sending in such great questions. And a big thank you to Rogelio. I will now pass the mic back to Jackie to introduce our next training speaker.
Okay, thank you. So the next speaker is Jayla Castan, who is currently a junior in the laboratory of Dr. Hilda Shervin. She is also a UCSF Propel Scholar. She is a native of San Francisco and graduated from UC Santa Barbara with a Bachelor of Science in Psychological Brain Sciences. Her work is a diversity supplement grant from the NIH. She has an amazing passion for science, and she plans to pursue her doctorate degree very soon. Additionally, Jayla is involved with numerous outreach initiatives, including College Track and the Emerson Collective. We are delighted to have her as part of our ImmunoDiverse leadership group. So please join us in welcoming Jayla as she tells us about her studies investigating the transcription factor Icaros and autoimmunity.
Thank you, Jackie, for introducing me, and thank you for the ImmunoDiverse team for having me today. This is my first presentation outside of my lab, so I'm really excited to be here. Like Jackie said, my name is Jayla Caston. I'm a part of the Shervin lab, and today I'll be talking about my preliminary data on this project that I just started last November, and that is categorizing the different immune cells that are infiltrating the lungs of our mutated Icaros mice. So to start off, what is Icaros? Icaros is a transcription factor that is crucial for the progression of hematopoiesis. It consists of 6 zinc fingers, where 5 and 6 are responsible for dimerization, and zinc fingers 1 through 4 are responsible for DNA binding. In previous studies, dominant negative Icaros has been categorized to behave similarly to Icaros null mice, and they usually have these zinc fingers 2 and 3 deleted. And when we see Icaros null and Icaros dominant negative in mice, there's this effect of no B cells and also some defects in other immune cells. So of course, not having this crucial element of adaptive immunity can cause serious disease. For example, in a patient study, we showed that there was this genetic mutation that occurred on zinc finger 3 that both P1 and P2 had. P2 was the offspring of P1, and P2 actually developed this child onset SLE, and P1 had no visible symptoms, but had immunological abnormalities similar to P2, where we can see here the total B cell numbers in both P1 and P2 were slightly lower compared to the healthy controls. So to further investigate this in our lab, we wanted to delete zinc fingers 1 and 4, since we know that zinc fingers 2 and 3, you get this complete loss of function in Icaros. So yeah, we were curious to see the different phenotypes when deleting both zinc fingers 1 and 4. And do these mouse models reflect any autoimmune phenotypes observed in human cases? So by conducting a Hep2 ANA assay, we saw that deleting zinc finger 4 had similar levels of autoreactive antibodies in their serum compared to the positive control, which is known for autoimmune disorders. And deleted zinc finger 1 actually had similar autoimmune or autoreactive antibodies in its serum compared to wild type and the negative control. So following this assay, we did a Immunohistochemistry staining, where we saw a significant increase of immune cell infiltrates in the lung of the deleted zinc finger 4 mice compared to wild type. And we see these beautiful structures of B cells, T cells, and myeloid cells that are infiltrating these alveolar sacs in the lungs of the wild type. In the zoomed out picture, you can also see these structures all over the lungs of these wild type deleted zinc finger 4 mice. So this led to my project to not only confirm what we see in immunohistochemistry staining, but to analyze and discover different categories of these immune cell infiltrates that are infiltrating the lungs of our mutant mice. So today I'll be focusing on the B cells that are infiltrating, and also within the subtypes of B cells, we'll be looking at follicular B cells and age-associated B cells. So my setup for this experiment is to harvest the lungs of the wild type and mutant mice at 5.5 weeks old. We process the tissue, stain with antibodies for flow cytometry, run these samples, and then analyze the data afterwards. So the first finding that I want to point you guys' attention to is this overall increase in total CD45 positive cells in our mutated mice. Although the percentages are average between both wild type lung and our mutated mice, we do see this overall increase in the significant increase in total CD45 positive cells. Next, I want to point your attention to the total B cell numbers, where we see a slight increase in total B cell CD19 positive B cells in the lungs of these mice, but I believe that is due to the overall increase in CD45 positive cells. But when looking at the percentages of these B cells, there's slightly lower amounts of CD19 positive B cells in the DF4 lung when compared to the wild type. And this reflects the B cell numbers that or the B cell percentages that we see in the blood of P1 and P2 in the human autoimmune study. Next, going into follicular and age-associated B cells, I just want to remind you all of B cell differentiation in both mice and humans, but I'm going to focus on the mouse markers in this experiment. So these follicular and age-associated B cells can progress after the follicular can progress after the transitional phase, and the follicular B cells and the age-associated B cells can develop after the mature B cell stage. And these are categorized as both being CD19 positive. Follicular B cells are categorized as CD23 positive and CD21 low, and age-associated B cells are categorized as CD23 negative and CD21 negative. So first, looking at the follicular B cells, we see a slight increase in the overall total B cell numbers. Again, I think that's associated with the overall increase in CD45 positive cells in the lung. But when looking at the ratio and the percentages, we actually see a decrease in the DF4 lung compared to wild type of these follicular B cells. And follicular B cells are associated with conventional adaptive immunity, and they've also been, lower numbers have been associated with autoimmunity and transplant rejection. Now going into the age-associated B cells, we see an overall increase in these age-associated B cells in both the total numbers and the percentages in our mutated mice. And this is very interesting because age-associated B cells, higher amounts of age-associated B cells have been associated with SLE, which is the disease that our P2 human study also had. So in conclusion, the preliminary data that we see is that there are more immune cells that are infiltrating our deleted zinc finger 4 mice than in wild type mice. We see less follicular B cells, a lesser percentage of follicular B cells in our mutated deleted zinc finger 4 mice versus wild type, and more age-associated B cells in our mutant mice. And so for future directions, alongside these B cell experiments, we've also been looking at T cells and myeloid cells. And I would love to continue looking at all these populations and their interactions with each other, as well as looking at the interactions between the immune cells. I'm really interested in looking at the immune cells' interactions with the lung stromal cells, and that idea developed from reading a paper actually from a lab here at UCSF, the SHUM lab, and how they saw an interaction with the immune cells and COPD patients with the stromal cells, and how they are interacting and creating cytokines to increase this infiltration into the lung. I'm also looking forward to looking at if these immune cells are creating any tissue damage in the lung. As we all know with COVID-19, the reason or why the prognosis is so bad is because these immune cells are actually attacking the lung tissues, and the lung tissues and also some other structures in the lung. And so I'm curious if these immune cells that are infiltrating our mutated mice are also creating this destruction in lung tissues. And that's it for me. I would love to acknowledge the Shervin Lab, for creating this home for me and allowing me to do experiments here, our collaborators that helped us with the tissue processing and some of the previous experiments. I would love to shout out ImmunoDiverse. They've really become my second family, and I'm so grateful to be a part of this team and to have so many wonderful postdocs and PhD students helping and supporting me along my scientific career journey. And of course, I would love to thank the NIH and UCSF for funding my project and allowing me to be here. With that being said, I want to thank all of you guys for your attention. And if there's any questions, I'm more than happy to answer.
Thank you, Jayla. We do have questions. First one, great talk. Is Icaros expressed in other cell types? And if so, do you think they could be contributing?
Yes, we have seen that Icaros is expressed in stromal cell types. I'm not sure yet how they could be contributing to this interaction between the immune cells and the stromal cells, but that is something that I would love to further investigate.
Great. Next question. Are there any pathological phenotypes associated with these changes in B cells you're seeing in the mutant mice, such as potential autoimmunity?
Any pathological phenotypes between the different deleted zinc finger 4 and zinc finger 1 mice?
Yeah, I believe so, and perhaps between those and the wild type. But yeah.
The wild type. Yeah, so we saw the HEP2 ANA assay where there were significant amounts of the deleted or autoreactive antibodies in the deleted zinc finger 4 mice compared to the deleted zinc finger 1 mice. The deleted zinc finger 4 or 1 mice, sorry, actually had similar levels of autoreactive antibodies in the deleted zinc finger 1 mice had similar levels of antibodies, autoantibodies as the wild type and the negative control. I didn't show this, but our H&E staining showed that the deleted zinc finger 4 mice had an increased amount of just cells infiltrating the lung compared to the DF1, and that's why we chose to go to do the immunohistochemistry staining with the deleted zinc finger 1 mouse and the not the deleted zinc finger 1 mouse, sorry, and instead looking at the deleted zinc finger 4 in comparison to wild type.
Great. Our next question is, great talk. Would you hypothesize in advance there are certain subclasses of T and myeloid cells that would specifically interface with age-associated B cells?
Yeah, that's a really good question. As of right now, I can't think of anything off the top of my head where they would be interacting. I'm sure that they are. We have been looking at the T cells. Yeah, we have looked at the T cells and like the differences in between the wild type and the deleted zinc finger 4 mice, and I can't think off the top of my head the stark differences that we have. Yeah, but I'm sure that they're interacting, and yeah, that's the next step of hypothesizing how they're interacting with each other and seeing these hopefully differences and levels between the deleted zinc finger 4 and the wild type mice, and then yeah, seeing how they're interacting with each other.
Great. Next question. Do you plan to do any functional challenges in these mutant mice to assess the relevance of the changes you're seeing?
Yeah, I have been thinking of doing a viral challenge. I'm not sure if that's the correct assay name, but yeah, I would love to see how these immune cells in the deleted zinc finger 4 mice are challenged with virus that are entering the lungs, that can enter the lungs of these mice. But yeah, that's a next step that I would love to do in the future.
Great. And just real quickly, before we go, I won't read the details here and we'll share this with you, but you've got a how would you feel about moving to Colorado question. to study vaccine-elicited T cell responses. So we'll share that with you separately.
Okay.
But thank you so much, JLo. That was a great talk, and we appreciate you sharing your time in science.
Thank you.
At this time, we'll go back to Jackie to introduce the next trainee speaker.
Yeah, thanks, Casey. Okay, so our next speaker is Lam Q. Fong, who is a postdoctoral fellow in the Department of Pharmaceutical Chemistry at UCSF in the laboratory of Dr. Bill DeGrotto. Her research focuses on understanding the biophysical forces that govern the structure and function of immune receptors. She has including the UC President's Postdoctoral Fellowship, a Ford Foundation Postdoctoral Fellowship, a UCSF Institutional Research and Academic Career Development Award Scholarship, and she is Howard Hughes Medical Institute Leading Edge Fellow. Please join us in welcoming Dr. Fong as she tells us about her fascinating work investigating the molecular forces under the structure of the NKG2C DAP12 receptor complex.
Hello, everyone. Can you hear me? Thank you for that kind introduction, Jackie. My name is Lam Q. Fong, and I'm a postdoc in the DeGrotto lab in pharmaceutical chemistry. I use she, her pronouns. And today I want to tell you about some of the work that I've been doing trying to understand the molecular determinants of assembly and specificity in multi-component immune receptors. So as you all know, there are many different types of immune cells and they perform very different functions. But something that's common among immune cells is that in order to perform that function, they rely heavily on proteins on their membranes to interact with the outside world. These proteins are in charge of performing two distinct functions. One is ligand recognition and the other is intracellular signaling. What's cool is that some receptors actually rely on two separate protein subunits in order to do this. So one of the subunits will do the ligand recognition and communicate somehow with the other and do intracellular signaling. This enables receptors then to have a response that is specific for a ligand, so you can change out the subunit while still conserving the signaling subunit. And one way in which this is conserved is through this Itam-based signaling. So what I'm showing here is a multi-component receptor, and what you'll notice is that these Itam signaling motifs couple to downstream phosphorylation mechanisms. Additionally, what you'll notice is that structurally, these signaling motifs have a large intracellular portion, while the ligand binding motif has a large extracellular portion. So really, the effectiveness of these receptors relies heavily on the interaction of these protein subunits in the membrane environment. One of the canonical multi-component receptors that you're probably very familiar with is the T cell receptor, which assembles multiple subunits into one T cell receptor complex. And this assembly relies on a conserved 2-to-1 acidic to basic motif, where basically you're bringing in some of the signaling motifs here shown with two aspartate amino acids to assemble with the T cell receptor with a basic amino acid lysine. And what we know is that in the absence of these polar residues, so if you mutate out an aspartic acid for alanine, you don't get assembly of the receptor complex. And so when you don't have assembly, then you don't have function in the receptor. What's cool about this motif in my mind is that it is conserved across multiple immune cell families. So here we have the Fc-associated receptors. And again, you'll notice this 2-to-1 association motif, as well as receptors that associate with a signaling module called DAP12. Okay, so at this point, I had a question, which is, okay, so these are required for assembly in these receptors, but these are polar residues that interact in the membrane environment. So what factors enable the stable arrangement of 3 polar residues in a non-polar membrane full of lipids? To do that, I decided to focus on these DAP12 associated receptors. I find these particularly interesting because DAP12 shown here in orange is this homodimeric signaling protein that assembles with over 20 different types of ligand binding receptors. Here I'm just showing 2 of them for myeloid cells as well as natural killer cells. And essentially, If you take a look at all of the, or some of the receptors that interact with DAP12, and you focus in on that membrane portion, what you'll notice is that there's not a lot of sequence similarity between the different types of receptors. The only thing that's conserved is that lysine residue that I told you was very, very important for assembly. So this really led me to the question, how does DAP12 associate with so many different ligand binding co-receptors? And in order to study that, I again turned to the DAP12 NKG2C receptor. So in this case, this is the only known structure of DAP12 with a co-receptor at the moment. And you'll notice here again that we have this 2-to-1 residue or polar residue motif. And what's interesting about these residues is under physiological conditions, aspartic acid can adopt a negative charge, whereas lysine can adopt a positive charge. So this led people to question, what is the potential protonation state of these residues? Because that could really influence its interaction in the membrane. And so under physiological conditions, we would expect that these would have a 2-to-1 negative to positive charge ratio, meaning you're bringing in a net negative charge into the membrane. But we hypothesized that maybe there were differences in the protonation state where maybe now, instead of bringing in a net negative charge, you might bring in a net neutral charge by protonating one of the aspartic acids into the neutral state, or maybe making it so that all of the residues are neutral. In order to test this question, we turned to a technique called molecular dynamic simulations, which many of you might be familiar with. And essentially, what we're doing with molecular dynamics is we're tracking the motions of atoms in our protein as a function of time. So you can imagine if you have an atom in your protein, we want to know how it moves in time. That happens based on the initial position of the atom. So you have to know where the atom started in order to know where it's going, the temperature of the system, which tells you how fast these atoms are moving, as well as interactions with other atoms. So for example, if our atom is positively charged, it might interact with a negatively charged atom in time by coming close together based on electrostatic interactions. So why MD is, in my mind, so powerful and so interesting is that it really gives us that atomic level detail that you need in order to assess how does one hydrogen atom affect the overall structure of a protein-protein interaction, as well as it gives us conformational dynamics about the protein. So in typical structures, we're usually stuck with like 1 or 2 structures of the protein. Here we can evolve the protein in time and get the dynamics. So our data looks a little bit like this, where you have this, again, DAP12 NKG2C receptor, this natural killer cell receptor in the membrane, and we can simulate lipids and water and all of the ions that we would find biologically. And we can do this as a function of protonation state. So I don't have too much time to talk to you about everything that we found, but here are some of the highlights. Essentially, if we look at this protonation state that had been widely discussed in the literature as how it could be existent in the membrane where we have 2 negative charges, one of the first things that we notice is that when we track the distance of these 2 aspartic acids in the polar residues, and we do a distance as a function of time, there's a lot of variability in that interaction. So what you have is these aspartic acids that are moving back and forth, essentially leading us to think that there was a lot of electrostatic repulsion, which you would expect because you have 2 negative charges close to each other. And so we can actually confirm this by looking at how in the membrane, you're pulling in sodium ions. So what this means is that you have 2 negative charges that are not compensated by 1 positive charge. So you need to bring in more positive charge into the membrane. Additionally, with all of that charge, you're bringing in these water molecules, which flood the hydrophobic membrane with water. This is especially stark when you compare to a complex 2, which is partially protonated. Again, here we have only a positive to a negative charge ratio of 1-to-1. And then this aspartic acid distance is very, very tight. Essentially, you don't have a lot of fluctuation like you did before. That's partially, in fact, in part because you don't have that electrostatic repulsion, but there's also the potential for a hydrogen bond now. Because you've added this proton, now you can hydrogen bond between the 2 of these, and you form a very, very tight hydrogen bond that we have seen, if you look at the proportion of time that it's persistent in our simulation, it's close to 100% of the time. So what that does for us now is it has these aspartates kind of positioned to interact very nicely with the lysine. And it forms this what I think is a very nice triangle of interaction that is very persistent in our simulation. So that means it's a particularly conformationally stable arrangement of the residues. And what you'll see here is that it's not that this is a static interaction. Certainly everything is moving around it, but it does maintain that hydrogen bonding network. And this is really enabled by that 2.5 angstrom hydrogen bond. So at this point, we asked, how do these locally conformationally stable dynamics influence the helical level of the protein? And so for this, we ended up looking at just the residues in the backbone and trying to assess what conformations are available to the helices, so the DAP12 and the NKG2C, and we grouped those into similar structures. So if you're a visual person like me, it would be helpful to just kind of look at what these groups look like. And essentially here, DAP12 I have in gray, and the NKG2C receptor is in color. And what you'll notice is that the biggest differentiating factor between these groups is the position of the NKG2C receptor. And when we look at the proportion of time these are explored in simulation, so this tells us about the stability of these conformations, it's pretty similar across the board. That means that there's no specific orientation that these helices adopt, even though they have a polar triad interaction that is very, very consistent and conformationally stable. So this for us really explains the ability of DAP12 to really interact with all of these receptors that are so sequence diverse, because it doesn't matter as much how the NKG2C receptor or any of these receptors lie relative to DAP12, as long as you're maintaining that very, very conformationally stable interaction with lysine. And so this is not something that, so DAP12 has a limit, and there are receptors that it can't interact with, and that's something that I'm actually looking into now. How does the sequence of DAP12 contribute to that to preventing that interaction? And so specificity. Oh, shoot. And with that, I'd like to thank all of you for listening. Particularly, I want to thank the ImmunoDiverse organizers. They've been just incredible in this whole process, as well as my mentors, Bill DeGrado and Michael Grabe, some of my collaborators on this work, Matt and Sophia, as well as many other members of our lab, and my funding sources. I'd be happy to take questions.
Great, thank you, LambQ. First question, you mentioned the TCR having similar polar residues in the membrane, but are there other molecules like DAP12 that form interactions like this in the membrane with so many different interaction partners?
So DAP10 is known to interact with a few, I think DAP12 is the most, I would say, ubiquitous in all of this that I've seen. The FC receptors has a couple of interaction partners as well, but I think why I focus so much on DAP12 is because it can interact with so many different things. And I thought if I found commonality there, I could then kind of extend it to these much more specific cases like the T cell receptor.
Great. Next question. Probably a naive question, but do physiological pH assumptions hold for microenvironments within the lipid membrane? How would this change assumptions going into the MD models?
Right, so the question is about whether we can say, oh, it has this pH in water, therefore in the lipid membrane where you're completely hydrophobic. It's no, absolutely not. I think that's why that question was so kind of relevant to me, because there are ways you can imagine where you have two negative charges being able to exist in the membrane. In some ways, a nonpolar environment enables the negative positive interaction to be stronger. And so that's why there's questions about this. But I think, you know, I went into it with the idea that it could be the doubly negative charge because it had been talked about so much in the literature and we really had no idea. But no, the pH considerations do not hold the same in the membrane.
Great. Next question. Very nice. Do you think that phosphorylation of the Itams in DAP12 could affect the protonation? Or does ligand binding to NKG2C affect protonation?
That's a great question. Potentially. So I started this project by working on a receptor called TREM2, which is on myeloid cells. And with that receptor has a lot of charges on its surface. And so you can imagine bringing a lot of charge from the part that binds the ligand close to the membrane, and that will absolutely change the environment, the electrostatic environment. So potentially, I haven't ruled out the idea that protonation is a way in which these receptors might contribute to assembly and disassembly in the membrane, but it would be really nice to get more like structural data of the full receptor complex in order to be able to study some of those effects. Sorry, that was a very convoluted question, but it's something that I've thought about.
Very interesting. I think really quickly we have time for one more question. Great talk. What do you think determines interaction specificity of DAP12 with its cognate receptors?
Okay, this is something that I'm sort of working on right now because there's this particular instance where DAP12 does not interact with NKG2D receptor. Well, it doesn't interact with the human one, but then it does interact with the mouse one. And there are aspects of the rest of the membrane sequence that contribute. And I think that there is, so I showed how there's like a helical dynamic. I think that there's a limit to how dynamic those helices can be. Additionally, it really depends. Well, okay, I'll just leave it at that. I think the dynamics of the helices actually really matter a lot. And what's cool is that simulation allows us to see to what extent this is dynamic. And when we go into sequences that are not as dynamic, then do we see differences correlating to experimental data that doesn't yield specificity.
Great. Well, sorry that we didn't get to all the questions. Thank you so much, LambQ, for that great talk. And again, I'll pass it back to Jackie to introduce our last training speaker of the day.
Okay, thank you. So finally, I would like to introduce Carlos Castellanos. Carlos is a PhD student in the BMS program at UCSF, working in the laboratory of Dr. Jung-Suk Shin. He's an alumni of the Initiative for Maximizing Student Diversity Fellowship from the National Institute of General Medical Sciences. Please join us in welcoming Carlos as he tells us about his exciting work investigating the role of MARCH1 in dendritic cells priming Th2 responses.
Great, Jackie. Thank you so much. It's such a pleasure to be with you all here today, virtually. I hope that you can see my screen and that you're not seeing the presenter view. So, and I just really want to thank the organizers of this ImmunoDiverse Colloquium. I just want to say that for me, it's been just an absolute honor to be part of the diverse community at UCSF. And I'm really happy to present this story. So I'm a 6-year PhD student, and the title of this is Lymph Node Resident Dendritic Cells Drive Th2 Cell Development Involving MARCH1. So dendritic cells are just a fascinating cell type, and they're involved in so many different aspects of immunity. And one of the parts that I'm going to talk about is their role in allergic sensitization. So we are all exposed to environmental allergens, and basically they get breathed in. These are animal, fungal, and plant products, and sampled by dendritic cells at the airway. And those dendritic cells internalize those allergens, process them, and present them on the surface onto these MHC class II molecules. So this process also somehow allows dendritic cells to undergo a maturation program, either directly or indirectly. which enables migration to the lymph node, that central center where T cells and dendritic cells meet for activation. So a dendritic cell may readily encounter that rare allergen-specific T cell and induce the clonal expansion and the polarization of that T cell to the Th2 cell lineage. And those Th2 cells produce cytokines like IL-4, 5, and 13, which induce allergic inflammation and asthmatic responses in some people. And so this is kind of a working model still, and we don't fully understand exactly the mechanism by which dendritic cells drive this Th2 cell polarization. So one prevailing hypothesis in the field is very quantitative and relies on the availability of peptide MHC class 2 on the dendritic cell surface. So more peptide MHC2 would lead to the Th1 cell fate, whereas low levels of peptide MHC2 would lead to Th2 cell fate. And I just want to emphasize that in order to get good TCR signal activation, you also need co-stimulation. So I'm also going to be talking about this co-stimulatory ligand CD86, which signals to the CD28 receptor. So this hypothesis brings up a very important question, is how would these be modulated in vivo by dendritic cells, and specifically for developing Th2 cells? So with that, I want to introduce MARCH1, which is a membrane-associated ring CH1 ubiquitin ligase. So it's a membrane-anchored ubiquitin ligase, and it's known to target both MHC class 2 and CD86 through its transmembrane domain. And then it allows the ubiquitination at the cytoplasmic terminus of those substrates to enable the lysosomal degradation. So one thing that is apparent in MARCH1 deficient mice is that because this regulatory mechanism is not occurring, they have a remarkable accumulation of MHC class 2 and CD86 on the cell surface. Now the mice are completely fine. They don't develop any sort of autoimmune disorder, but it's kind of intriguing that they have this like very high level of these two very immune activating ligands. So I wanted to ask whether MARCH1 was playing a role in immune activation and specifically in allergic airway inflammation and also Th2 cell differentiation. In order to do this, I used a very physiologically relevant model of allergic asthma in which I sensitized the mice with house dust mite, which is a clinically relevant allergen, using this protocol here. And what I observed was that MARCH1 deficient mice had a remarkable absence of Th2 cell development in the lungs. They didn't recruit those IL-4 or IL-13 producing CD4 T cells. They also had very, very low eosinophilia. So they didn't recruit these other granulocytic cells also associated with allergy. And they also failed to mount IgE antibody responses. And IgE is a canonical antibody associated with allergy. So all of these immune reactions help to facilitate the pathology you see in people with asthma, including the narrowing of the airways. And we can actually model that in the mice using airway hyperresponsiveness. And in MARCH1 deficient mice exposed to HDM, we observed that they didn't elicit airway hyperresponsiveness. However, wild type mice exposed to HDM did. So this told me that the airways in the wild type mice were impaired. And indeed, if we look by histopathology, looking at goblet cell metaplasia, these are mucus producing cells, the wild type mice had more mucus producing cells along the airways than the MARCH1 deficient mice. So this told us that MARCH1 was important for eliciting Th2 inflammation and the pathology associated with asthma. So then I wanted to ask whether this was happening through dendritic cells. And so I used the Krelock system using mice in which MARCH1 was specifically deleted in dendritic cells using the C11C cream driver. And those mice fully recapitulated the MARCH1 deficient mice. They had fewer Th2 cells in the lung, fewer eosinophils, and they had severely dampened IgE antibody responses. So this also said that dendritic cells were playing a role, that MARCH1 in dendritic cells was playing a role. So I wanted to know whether this was occurring in the lymph nodes, since dendritic cells have been implicated so much in inducing T cell immunity in the lymph node. And indeed, when I looked in the lymph node using a sensitization protocol and just sensitizing the mice 3 times, I observed that the mice lacking MARCH1 in dendritic cells had much fewer IL-4 producing T cells in the lymph node. And just to further emphasize that this was a dendritic cell specific phenomenon, when I did the same experiment in mice lacking MARCH1 in macrophages, I didn't observe this defect, or in mice in which the MARCH1 deficiency was confined to either plasmacytoid dendritic cells or B cells. So this told me MARCH1 in DCs is required for inducing Th2 inflammation to allergens. And for the remainder of this talk, I'm going to focus on the mechanism. So one thing that I observed was that MARCH1 expression in different dendritic cell populations seems to vary. So in the dendritic cells at homeostasis in the lung, there's appreciable levels of MARCH1, but in migratory dendritic cells that then go to from the lung into the draining lymph node, MARCH1 expression severely is downregulated, and that is both at steady state and after allergen challenge. And one thing that was also intriguing was that there are these lymph node resident diseases which never see the airways. They develop solely in the lymph node, and those are the highest cells that express MARCH1. So then I wanted to parse out the contribution of MARCH1 among these different dendritic cell populations. And I first addressed migratory dendritic cells, knowing that CCR7 is this chemokine receptor crucial for directing dendritic cell migration from tissues to lymph nodes. So in mice in which the only dendritic cells that were capable of migrating were MARCH1 deficient, I didn't observe a deficit in Th2 cell development in the lymph node compared to the wild type mice. So this really told me that migratory DCs were probably not playing a role in Th2 cell development. So I wanted to address the role of lymph node resident DCs. But in order to do this, I run into a big problem because lymph node resident DCs would have to activate T cells before migratory DCs get there. And that's during the very early T cell activation events. So I have to look extremely early when T cell immunity is occurring before migratory DCs get there. But those allergen-specific T cells are extremely rare. So I used an adoptive transfer strategy to be able to enrich for allergen-specific T cells using T cells that are specific for the model antigen ovalbumin. And then I challenged the mice with ovalbumin and house dust mite. And I just wanted to make sure that this recapitulated the phenotype I observed polyclonal, and indeed it did. So those mice which received OT2 cells that lacked the MARCH1 in dendritic cells, those OT2 cells produced much less IL-4. So now I have this interesting system. So now I needed to block dendritic cell migration. And in order to do this, I used this pertussis toxin, which blocked dendritic cell migration, as you can observe here. So one thing that I observed was that when I blocked dendritic cell migration, I actually did not observe any deficits in T cell activation, both by NUR77 or CD69. And most importantly, polarization of T cells occurred just fine based on the canonical transcription factors GATA3 and TBET. So this told me that dendritic cell migration is likely not playing a role in T cell activation and polarization, at least in this system. So then what about in the context of MARCH1 deficiency? I could observe very early as 4 hours, adequate T cell activation of T cells in both wild type and MARCH1 deficient mice based on NUR77. And they displayed similar kinetics of CD69, although in the MARCH1 deficient setting, CD69 was retained in the OT2 cells. And most importantly, GATA3, the transcription factor that guides Th2 cell development, was not induced as robustly in the T cells transferred in MARCH1 deficient mice, and instead those T cells were making more TBET. So this told me that the MARCH1 mediated Th2 cell development was occurring not through the migratory DCs, but through the lymph node resident DCs. And just for the last 2 slides I have of my presentation, I'm going to focus on the mechanism. So I looked at the mice in which MHC class II could not be ubiquitinated, and those mice recapitulated some of the phenotypes that I saw in that OT2 transfer system, but not all. In mice in which C86 could not be ubiquitinated, I didn't see any major differences in that OT2 transfer system. But it wasn't only in the mice that both MHC class 2 and CD86 could not be ubiquitinated, which recapitulated the March1 deficient mice, in which I observed that those OT2 T cells retained CD69, had less GATA3 and more TVET. And indeed, those mice also exhibited a similar deficit in Th2 cell induction, both in the lymph node, in the sensitization protocol, and in the lung, in the airway inflammation protocol. So this tells me March1 mediated class 2 and C86 ubiquitination by lymph node resident disease promotes the development of Th2 cells and subsequent allergic inflammation. So I just want to bring up this model again and say that this is not the only model to explain how Th2 cell development occurs. We should also consider this model in which allergens can readily penetrate and get into the lymph node, either through the lymphatics and access the lymph node resident DCs. And through a March1 dependent mechanism, these lymph node resident DCs guide Th2 cell development. And so in support of this model, there's a lot of literature, including work from Ron Germain that says that TCR signaling strength is a major determinant in Th1 and Th2 cell fate. And March1 would be a way for dendritic cells to regulate the amount of TCR signaling strength. Also, patients with atopic disease often show mutations in TCR signaling. And so the T cells in those patients are getting less signals and they're more likely to become Th2 cells. And we're not the first ones to show a role for lymph node resident DCs in T cell activation. Work from Ron Germain and from Mark Jenkins has also shown a role for these DCs in the lymph node. And just to leave you with some food for thought, the Th2 cells in humans with allergy harbored the preferential specificity for the small and soluble antigens in the allergens that are more likely to penetrate deeply and get into those lymph node resident DCs. And just to leave you with a final question, I think my research brings up the question whether targeting March1 might be beneficial and a promising strategy to treat the allergy patients that are at risk of developing asthma. And with that, I would like to thank my PI, Dr. Young-Suk Shin, who has been absolutely a fantastic mentor to my entire PhD, as well as all my amazing collaborators here at UCSF and my funding and support and my thesis committee. And I welcome your questions. Thank you.
Thank you, Carlos. Super interesting work. And I liked the question you posed at the end. I was wondering about that myself. We're a little short on time, so I'll maybe just ask a couple questions and we can do a couple brief answers. So first question, great talk. Can you elaborate how you disentangled the role of DC versus macrophage March1 deficiency using the CD11C CRE model, considering that the alveolar macrophages express CD11C?
Right. So in data I didn't have time to show. I also used another more specific dendritic cell CRE driver. It's called the CBTB46 CRE. And I could also corroborate the findings with the CD11C CRE mouse. And moreover, I used the MAFB CRE mouse. And so those mice, you're targeting just the macrophages. And in those mice, I don't see a deficit in Th2 cell development. So that tells me that this is happening through the conventional dendritic cells.
Great, interesting. And just one last quick question. What about non-allergen models? Do you think the same pathways are employed for Th2 responses to infections with worms in the gut?
Yeah, so this is also a very interesting question. And I actually did infect the mice with Nipostrongylus brasiliensis, which is a classical worm model. And they also have a remarkable deficit in Th2 cell development in the lung. I didn't actually look in the gut, so I'm not 100% sure in that case. But I think we need to think about whether the allergens are actually, or the antigens in this case, are actually getting through the lymph node, either through the migratory DCs, or they can penetrate readily and get into the lymph node through the draining lymphatics and into the lymph node resident DCs. And so I think it's context dependent for sure.
Yeah. Well, thank you so much, Carlos. It was a pleasure hearing about your work. And With that, I will send it back to Jackie to take us to the break.
Thanks, Casey. So now we're going to be taking a short, quick break, and then just make sure you come back and don't miss out of the panel Q&A with our 5 speakers. And then we will also have our presentation by the ImmunoDiverse Allyship Group, and we'll be announcing the Community Award, and then we will have our final talk by our social keynote speaker, Dr. Dabrowski, followed by a group discussion. So see you all in 3 minutes. Thank you.
I think our break is over. Thank you so much for staying on. I hope you're enjoying our programming, and we will start to move on to the panel Q&A with our amazing trainee speakers. We've all gotten a chance to hear about their work, what they're currently doing, and in the next 20 minutes, we will get to know a little bit more about them, the backgrounds, and a little bit about their experiences as trainees. Okay, while we get everyone spotlit, I'll get started with the questions, but I'll first actually introduce myself. My name is Priscila Muñoz Sandoval. I am a third-year graduate student in Mark Ansel's lab. I am also an HMI Gilliam Fellow and chair of the high school outreach. To get us started, I would like to start us off by asking all of you if you could please tell us a little bit about your backgrounds, where you're from, if you're the first to pursue a degree in your family, especially in science, and kind of what led you to this path that you're on. We'll go ahead and start with Carlos.
Great, thanks, Priscilla. So yeah, my name is Carlos, and I'm originally from Colombia. So I was born in the South America, and I moved to the United States when I was about 9 years old. Yeah, so I'm an immigrant basically, and I fully identify kind of as like you know, as somebody very diverse that brings like a lot of diversity to to a lot of places. And what I mean, my family. So my family had to make a giant sacrifice when they moved to the United States. My dad was a medical doctor, and when we moved here, he couldn't practice medicine. And my mom too. She had to learn English and become a nurse and everything. I was always very much interested in like the medical field, and I thought, oh, I'm gonna pursue their footsteps and go into med school and all this. But when I started going into high school and college, I became way more interested in the knowledge and the fact that there were still a lot of things that we just didn't understand very well. My dad was an OB-GYN doctor in Colombia, and one thing he told me was like, yeah, preeclampsia is this one disease we don't fully understand, and we don't even know how to treat it. And so when I went to MIT for a degree in biological engineering, and I got exposed to the research, and that's when I decided I want to become a researcher. And my path has led me into immunology, and it has just been amazing. And I like really love the work that I do, and I want to continue doing it.
Thank you, Carlos. And we'll actually go in reverse order. So, Lamku, same question.
Hi, yeah. As I mentioned, my name is Lam Q. I was born in Cuba, and I immigrated to the US at the age of 7. And so I lived in Los Angeles, and then we moved to Puerto Rico, and then I lived in San Diego, and we just moved around a lot when I was a kid for no particular reason. Honestly, I'm not really sure why. But my interest in science really fully developed actually in college. I thought that I wanted to be a lawyer when I was a kid because I asked a lot of questions, and someone just said, you should be a lawyer. Like, that's what lawyers do. They ask a lot of questions. And so for a long time, that's what I thought I wanted. But ultimately I ended up pursuing a degree in chemistry at UC Berkeley. And there I didn't realize what chemistry was, to be honest, until I got there. And it really wasn't until I got into laboratory setting later on in my junior year that it actually dawned on me, like, this is exactly what the questions that I was asking before are leading me to do. And so I did a lot of physical chemistry. I did my PhD in physical chemistry at Northwestern, but I was always kind of immunology adjacent. Like, I've taken a good number of classes. My brother's a type 1 diabetic. I'm a type 1 diabetic. There's a lot of immune related things that are part of my life and my trajectory. And so that's kind of like what's led me toward a more biophysical field, because it really allows me to bridge this like chemistry and this thing that I learned and I really grew to love with something that matters a lot to me, which is how does the immune system work? How does it respond? What happens when it goes haywire? And I mean, my parents are immigrants. I think that everywhere you go, like you kind of feel like an outsider. But at the same time, you know, I think the mentorship that I've received along the way has been very, very crucial. And it's like, you know, one person here and there who just really tells you, like, they super believe in you and they tell you, you know, you can do anything. You know, you go into a lab and you synthesize a bunch of particles and you have no idea what you're doing, but someone tells you, you did an amazing job. And I think that that's what has enabled me to continue to pursue this career, even though oftentimes I find myself being the only one in a room, the only one who signed up for, you know, that can be a very big challenge.
Thank you, Anku. Jayla?
Hi, everyone. My name is Jayla. I'm a San Francisco native, born and raised, actually second generation San Francisco native, specifically from Bayview's Hunters Point. And if you guys are not from San Francisco, this is a historically marginalized region of San Francisco. It has historically been redlined, and that is why I grew up there. My grandparents were only able to buy property in certain areas of the city, and Hunters Point was one of them. Growing up, I would be really ashamed to say that, but now at the place that I am now, and now being able to outreach to this community, I'm so proud that I was born and raised in that community that really raised me and pushed me to go against all odds and yeah, just be an advocate for my community. How I got into science, I've always been interested in science, always loved it, loved asking questions, loved the problem solving aspect of science and math, always did well in those classes. So naturally, well, the only thing that I was told that I could do with science was becoming a doctor. So when I set out for undergrad studies, I was like, I'm gonna be a kinesiology major and be a sports doctor. That didn't really work out too well. I saw, you know, someone playing basketball and their shin going straight through their skin. I was like, I can't do that. And when I transferred from the school where I was doing kinesiology to a city college, I was like, okay, I want to go to UC next, and I really want to do biology. But every time I talked to a counselor about me wanting to do biology, me wanting to be a doctor even, I was heavily strayed away from that career, always being told, no, you can't do that. You're not going to get into the classes. You're not going to do well in the classes. This is just something that's not for you. And I was persuaded into studying psychology because it was easier for me. Although my peers in Santa Barbara, mostly white males who were doing biology, they didn't get the same criticisms from the same counselors. But yeah, so I got into psychology, and luckily that's where I fell in love with studying the brain and learning about the brain. And then going into a UC system my junior year, I found out about research and how research is basically getting paid to be curious and to learn for the rest of your life. And I was like, that's the job that I need. And yeah, I've been trying to be a researcher ever since I learned about it about 2 to 3 years ago now. Actually, sorry, 3 years now. Yeah, and I'm so happy that I pursued this career, getting a job at UCSF and now being in an immunology lab. To me, it kind of shows that although these counselors and people were doubting me and saying that I cannot do biology, it's kind of like destiny, fate, if you believe in that I ended up in a microbiology lab, something that I was initially so passionate about. I ended up where I wanted to be. And because I'm in immunology now, I'm really interested in the immune system and would love to see how the immune system and how our immune system and neurology kind of interact with each other, and just discovering that more, hopefully in my PhD studies.
Thank you, Jayla. I love to see all the passion everybody has. Rogelio, same question.
Yeah, thank you. I am originally from Mexico, and my grandparents and my ancestors are indigenous people from the south of Mexico. I grew up in Oaxaca. My parents were the first in the family to go to college, but I am the first scientist in the family. I became interested actually in science and research in high school. I had opportunity to participate in some science Olympiads, and yeah, I changed kind of like the idea that I had of, oh, I maybe want to be an architect, or to, I can do research, and you can go to lab and you can do experiments. And yeah, in a matter of probably 6 months or so, I changed completely the idea I had for a long time. And because I had the opportunity to go to the university while I was still a high school student, no, so I went to college in Mexico City, and then I did my PhD at Harvard, and now I am here. I started as a chemist, moved as a biophysicist, and slowly moving into biology. I feel that biology, and in particular immunology, is just amazing, no? And yeah.
It's true. It is amazing. Gail, same question.
Hi, so I'm Gail. I am actually an immigrant as well. We moved here from the Philippines when I was a teenager, so not too far from Carlos' experience. And we actually moved to Texas first, where I was one of the only Asian students in my whole entire school. So that was very difficult, and it was a huge sort of culture shock to me. So when we finally moved to California, it was it was a lot more diverse. So I'm very happy in California and would want to stay here for a while. Yeah, so I did my undergrad at CSU. It's a primarily sort of teaching institution. We don't really have that much research. So the first time I got exposed to research in that sense is sort of at UCSF, actually, through the SRTP program, which is an amazing program, and I hope they keep doing it starting this year, since I'm positive that we can keep doing research again in person, and hopefully we can bring in undergrads again. And I actually got interested in science from a very early age as well, because my grandpa, who barely had a high school education, was very well read, and he was actually super into science and wanted me to be a doctor, to be the first doctor in the family, which unfortunately did also not pan out, because I was terrified of the ER where I volunteered at the at the unfortunate wishes of my dad, who wanted me to be a doctor. But yeah, so my parents were also the first to go to college, so I'm not the one who's first generation, but I will be the first scientist, I guess, when I graduate. So that will be nice. Yeah, and immunology is such a fascinating field. I honestly didn't even know what immunology was until I did my internship at UCSF, and have been in love ever since. So.
Thank you all for sharing your trajectory. I wish we had more time to delve into a few more things, but you all have unique stories and unique perspectives that you bring to science. And in the last few minutes that we have, I actually want to move on to probably the last question that I had and skip over a couple and ask, in your opinion, what are the most impactful ways faculty can affect change at their institutions in matters of diversity, equity, inclusion, and justice, since I think mentorship seems to be also a trend amongst all of you. And with that, I'll actually hand off to Jayla first.
Cool. Well, I think that I don't think that there's a best way for anything, but one way that I would suggest is, I'm so incredibly proud of the allyship team that is a part of ImmunoDiverse. And I think one way faculty can get more involved is being a part of these allyship meeting discussions, learning about the socioeconomic injustices, about the structural racism that is affecting our whole system, and not just looking at it in a university, but also looking at it from the structural racism that is effective in our whole government, that is seen throughout all of society. I'm really appreciative of the faculty that has been a part of ImmunoDiverse and has been going to these allyship meetings and being a part of this change. I just hope that this work continues. I think we all know as scientists that in order to make a impactful, significant contribution, there has to be repeats. It's not just a one and done experiment. It has to be repeated over and over and over again until we get it. There has to be troubleshooting. It's not just one answer and then you're done. So I think we need to apply what we know as scientists to attacking structural racism within our science community, and also in the larger spectrum outside of the science community, within your own communities. So yeah, I just really encourage you all to continue to fight, continue to learn, continue to advocate. This is not just a one and done. This is not just a trend that universities should be following to let's all increase diversity now. This is a fight. This is a struggle. We should look at it just like how we look at experiments and being making contributions to the science community as a whole. Yeah, that's kind of my 2 cents on it. Yeah.
Thank you, Jayla. Anybody else would like to add on to that?
I just want to say maybe that, like, yeah, I totally agree with Jayla that I think we should be doing experiments, no? In the sense of, like, I think there is a lot of things that we can try, and there is a lot of ideas out there. And I think I'm really glad that there is events like this happening, but like all the entire initiatives. And I think, again, also at all levels, we have our thing to contribute, as a faculty, as a trainee, as a student. I think we should all be involved, be part, and talk about common things. I feel that there is a lot more than we realize that we share, and that could be kind of like the starting point to think how we move forward.
Carlos, you had muted yourself. Did you want to add something?
Yeah, I think that faculty can really help by really listening and by really being there when it really means a lot to us to be there. And that's what I've had from my PhD mentor. She's just an amazing listener, somebody that just internalizes what I say and really has a great response to it. And I feel like that this is a problem, that people don't listen, that they don't understand what we're saying. They just say, you guys figure it out on your own. And this is something that they can, like, if we all commit to just understanding where we are coming from, I think that that's going to be very important for actually making some change. So
thank you, Carlos. I wish we had time to hear from all of you. You're all amazing scientists and your trajectories are inspiring. I'm inspired in the few minutes that I've talked to you. Thank you all for being here. Thank you for pushing forward. For those of you that are already past on your postdoc and moving on to your next steps, I wish you all the best. To Gail and Carlos that are just finishing up. Also, I can't wait to see what you're going to do. And Jayla, I think you're going to do amazing things. So thank you. And with that, I will transition us to our allyship presentation with Joelle. Thank you again.
Thank you.
Thank you.
Hello, everyone. I'm very excited to see so many familiar faces and also unfamiliar faces here. I am Joël Babdor. I'm postdoctoral researcher in the Spitze Lab at UCSF, and I am the chair of the allyship learning program that I'm going to talk about today. I'm going to share my screen. Sorry about that. Here we go. Can everybody see my screen well?
Yeah, we can see it.
Awesome. So sorry about that. Victor and Elizabeth have already mentioned what ImmunoDiverse as a whole has as a mission, and I'm going to specifically focus on one of the key initiatives that we have set out to tackle, which is the community education aspect of our work. And so to do that, we have created a liveship learning program. And I'm going to tell you everything about it. But first, let me lay out the plan for this discussion. We're going to start by just asking the question, what is a liveship? And I think it's an easy question, but it's actually maybe not that easy. And then we're going to talk about what a liveship learning, how a liveship learning is actually a very familiar process for scientists. And I'm going to try to convince you that this is something that we know how to do. And then I'm going to share about the social experimenting that we're leading here at ImmunoDiverse with this liveship learning program. So first, let's start with this question, what is a liveship? And I think, it could take many forms. Everybody would have a different perspective on that. So what I do in general when I have a question and I want to learn about something, I just Google it. As a good scientist, finding resources. And actually here, it's not science resources, but just random website that I went on. And what I've seen is the first resource here, a website where the definition of a liveship is actually a negative definition, which is interesting. And so here, a liveship is not an identity, but it is a lifelong process of learning and unlearning, building relationship based on trust, consistency, accountability with marginalized individuals and groups. The second point of this definition is that a liveship is not self-defined, and that our work and efforts must be recognized by the people we seek to ally ourselves with. I went on to another random website on the Tulane University School of Social Work, not so random, I would say, where here the definition of being an ally is to engage in self-examination, seek information to increase awareness, and find ways to make a positive impact. And I just continued like that. And here is a third website with a funny logo, Building Allies. And in their definition of being an active ally, and I like the fact that they're specifically mentioning active, which is very important. The definition is an active ally is someone who witnesses injustice and responds to it in any situations with actionable awareness. And so basically taking all these different definitions, then we can go on and find even more perspective on what is allyship. It seems that an ally is someone that consistently questions themselves and recognizes their lack of knowledge and therefore engage in a lifelong learning process. And it's actually interesting because to me, it sounds like it is exactly what scientists and researchers do, right? So we start by recognizing that we don't know something about, a biological question. So we recognize our lack of knowledge. And then we constantly question ourselves, making experiments, is asking all these questions, and we just keep asking questions, right? And then because we want to understand and learn, we engage in this lifelong learning process by reading all these papers and all this literature about this one specific subject or many subjects. So basically, everything that liveship learning requires seems to be things that we know really well to do in our scientific life. And I think Jayla and many other people in the panel have highlighted that, you know, this is something we know how to do. So potentially we can do it, right? But then, you know, it's the fact that we know how to do it doesn't mean that it's easy to do. And so we've been trying to think, you know, critically, what is the best way to go about allyship learning for our community? And, you know, there are many resources and one can get easily lost, and we could, you know, send long lists to the whole community of resources. And you may have seen one of these anti-racist resources, you know, in the last 12 months, but where to start? And even reading one resource, many people would have very different perspective, and one would just interpret what they're able to. And so basically we've been trying to think about how we can try to overcome these challenges. And the answer that appeared to us is that the community is actually the answer of all these challenges. Learning allyship is much more powerful as a community, and because everybody has a different perspective. And, you know, reading a resource or learning about a resource, the magic actually happens when we're able to share these resources and these different perspectives and reflections. So we've decided to create the allyship learning program as a community learning process. And here is the allyship learning program, but that's a complex image. Let me walk you through the different components of the allyship learning program. First, we're gathering information around a monthly theme. And so we've been trying to be mindful of really starting from the basis. And I'll share the different themes that we've decided to start with in a minute. But around these monthly themes, we want to basically curate a limited number of resources. And we want to make it, you know, accessible, and different learners learn different ways. So we have, you know, multiple resources that are of different format. And so we have an article, always a video, and also a podcast. So we can really, you know, take a theme and a question and learn in different ways. And these are the themes that we've decided to tackle for our first 6 months of the allyship learning program. And so we started by building community for January, which is the basis. We really wanted to be able to understand what it means to build community. And we went on to ask the very important question, what is systemic racism? And so, for example, we used the 13th documentary of Avad Verney to try to tackle this question, but also other resources. The month of March was focusing on what is intersectionality and why is it why is it important to account for it? And we've been using, for example, They Don't Say Our Names Enough from the Code Switch podcast. April is Racism in Science and Medicine. May will be What Are Systemic Barriers in Academia? And June will be What is Allyship? We want to go back to that, you know, initial question. What is allyship after, you know, 5 months of learning different aspects of that? And so beside these resources that we share with the community, we are also curating monthly learning events. There are so many events going on that we actually decided not to create our own events, but actually to piggyback on some of the events that we've seen in our community at UCSF, but also around. And so we've been able to partner with you know, these different programs, actually most of them at UCSF. And so for the month of January, we've been having a UCSF Restorative Justice Community Circle that has been very empowering to set the basis on the way that we're going to go about tackling all these very difficult questions as a community. The month of February has been for the question of what is systemic racism. We had the chance that Carlos Hoyt was presenting to the graduate faculty development program about the role of science and scientists in combating racism. And for the month of March, we've partnered with the Binyup Center for Microbiome Medicine and encourage our community to attend the Anti-Racism Book Club with the book of Audre Lorde, Sister Outsider, which has been very interesting to read. And for the month of April, we have the chance that the UCSF School of Medicine is developing a full event series on racism and race, and we're going to encourage the community to attend these events. So basically, just for us looking around and seeing, these different events, learning events that, the community can attend has been a very interesting process, and we've been sharing that with the community. And finally, the last component of the Leadership Learning program is the small group discussions that we're running at the end of the month, after people have a chance to go through the resources, go to the monthly events, and basically process all this new information, or not necessarily new, but process again some information. We wanted to have a space where we can gather and discuss these different resources and share our different perspectives on these different questions. And basically, what we want people to, get out of the group discussion is basically two things. On one end, it's learning our starting place. What are our biases, our privileges, and the preconceived notions that we have about race and racism. Also understand our own contributions in systemic racism. And the group discussion here reinforces this aspect of developing awareness and reflect on new ideas and concepts, because we are sharing all these different perspectives. On the other side, the other goal of the Leadership Learning program and the group discussion is we want people to learn how to be better allies, and basically using our privileges to amplify voices of underserved groups, own our own mistakes, and learn how to reduce harm. And basically from there, using the group discussion to share action items at multiple levels of our lives. And Carlos Hoyt was showing or describing those three different levels: the macro level, which is very large society; the meso level, which is a smaller group and community around you; and the micro, which can be your own individual or very small setting around your individual. And I want to share here some of the types of action items that are shared during our group discussions. And it is so rich that, I just couldn't put everything in there, but it seems to all, cluster around four main action words. The first is to join, and joining discussions about race and racism is very important. And so here, the action items, if you're at UCSF and you're not part of the Leadership Learning program, join the Leadership Learning program. If you're not at UCSF and in your institution there is a similar group, join this group and have this discussion. If you don't have any group in this, that proposes this sort of collective work, please reach out. We want to share our recipe. We want the one thing that we want is that this model of the Leadership Learning program to spread across UCSF and in academia in general. Another set of action items is to normalize discussions around VI issues, and we can actually think of normalizing this at different levels, in lab meetings, in research group meetings, at the lab retreats, at the department retreats, basically in the different places where we gather to discuss about science. We want to normalize as well discussion around diversity, equity, and inclusion, because this is going to be the place where we're going to be able to share. The other type of action item is to amplify, and we want to emphasize amplifying URM scientists, nominate URM scientists to seminar series, panel discussions, steering committees, awards. This amplification is very important. And the last aspect of action items that people can take and that we've been sharing about during our group discussion is to advocate within your institution, and basically with the goal to diversify at all level, at the faculty, postdoc, grad student level, and ask the institution to collect really demographic information annually and to basically start recruiting more underrepresented minorities so we can have a more diverse community and a more powerful community. Another thing that we want people to take from being part of the Allyship Learning Program is to feel more confident and more strong into being more active. So we want people to take the Allyship Learning and to move it into Allyship in action. And for example, you may not feel comfortable marching in the streets, or you may have not felt comfortable in that, but for some reason, we want the Allyship Learning to be a step where we can understand some dynamics and feel more confident to be part of some of the actions. Other types of actions are the outreach programs, and some of the speakers today I've shared about their own initiatives. ImmunoDiverse is creating multiple outreach initiatives, and so please tune in if you want to know how you can be part of this Allyship in action. So the Allyship Learning Program has been running for 4 months now, and here are some of the way that we try to analyze the exposure that the community has had. And so basically we've collected the information of 64 different individuals that have participated in our different group discussions. Obviously, we can't really measure how many folks have read the resources. We can only measure the people that are attending the group discussion. And we have a breakdown here where you can see that we have a pretty balanced number of grad students, postdocs, faculty, staff. And so we're very encouraged to see that in all these different groups, we have folks in the community that are joining the discussions. And it's been actually something that has been very important during the group discussion to have these different perspectives and have been a source of a lot of learning, really important learning. And before to leave you, I want to emphasize and acknowledge the group of individuals that are on the day-to-day making the Allyship Learning Program happening, and the group of people that are making sure that the vision is translated into a real Allyship Learning Program space for our community. And so all the individuals that are here on this picture are on the daily basis, gathering and creating resources, creating promotional material to advertise the program, preparing group discussions, showing up and moderating those group discussions. And really every individual has been contributing, and it really takes a whole village to make such a program happen and continue to run as successfully as it has been running. And I'm deeply grateful for their work. And I'm gonna just leave you there with what is coming next. We're gonna have an ImmunoDiverse Community Award, and then there's gonna be the social justice keynote talk from Dr. Dabrowski-Herbert that I really encourage you to attend. It's gonna be, I know it's gonna be fantastic already. And we'll follow with an ImmunoDiverse Colloquia group discussion. We want you to have a taste of what the group discussion looks like. And we're going to focus on sharing action items about how to create a more welcoming and supportive environment for URM colleagues at different levels of our lives, the macro, meso, and micro level. And I'm going to pass the microphone now to Oscar that is going to lead us to the next part of the program.
Can you all hear me? Hi, everyone. My name is Oscar Aguilar, and I'm a postdoctoral fellow here in the laboratory of Professor Louis Lanier. I'm also a Cancer Research Institute Irvington Fellow and hold a PDEP award from the Burroughs Wellcome Fund. And within ImmunoDiverse, I play the role as the chair of the outreach team. So in addition to honoring the scientific talent of our URM scientists, we feel that it's equally important to highlight those individuals that have pioneered the way for our community. Therefore, at this point in our colloquium, we would like to take a moment to honor those individuals, two individuals specifically, that have been instrumental in laying the foundation for our ImmunoDiverse program. The first individual has been an advocate for folks in the DEI space ever since she joined us from Vanderbilt University. She is involved in coordinating UCSF's Summer Research Training Program, or SRTP, which provides opportunities for undergraduate students to train in our research laboratories. She's also involved in overseeing the National Institute of General Medical Sciences Initiative for maximizing student development, which is a program to improve the academic and research competitiveness of URM students in the biomedical research field. This past year, she helped the Black students at UCSF form a community known as Black Excellence in STEM, or BSTEM. And as if this wasn't enough, she has also been generating anti-racist and DEI leadership courses and materials for the UCSF community. On top of all this, she has been a source of support for URM students in the BMS program as they navigate the academic arena. This all while dealing with the additional emotional stresses that this past year has brought. She is truly an inspiration for our students. The ImmunoDiverse team looks forward to working with her on training programs in the future. She is the Assistant Dean for Diversity and Learner Success, and also an Assistant Professor here at UCSF. Please join us in congratulating Dr. Deanne Duncan.
Congratulations to the ImmunoDiverse Colloquia planning team on a successful first annual event. Thank you so much for honoring me today as one of your first recipients of your community award. It means the world to me that you all as students and postdocs have selected me, and I'm so grateful that I get to work with wonderful students and postdocs every day to cultivate a healthier and more equitable, inclusive training environment for all of you. I hope that you all enjoy your first annual event, and I am thinking about all of you while I am desperately resting today. So thank you so much, and I hope that you have a successful event.
Thank you, Dr. Duncan. So secondly, we want to take a moment to highlight one of our very own. This individual is a recent graduate of the BMS PhD program, where she worked in a laboratory of Dr. Matt Spitzer, investigating the impact that bearing a tumor weighs on the systemic immune system. During her time at UCSF, she was also involved in organizing IG Equity, formerly known as ImmunoXX, which is a trainee-led group aimed at tackling gender equity in science and immunology. In the past year, as she was extremely busy wrapping up her PhD studies, writing her thesis, and pumping out a phenomenal article in Nature Medicine, she also poured all of the energy that she could spare to help create what is now ImmunoDiverse. She knew that she was not going to see or experience the fruits of her labor, yet she selflessly volunteered her time to ensure that our group would provide an inclusive environment and would be a sustainable program. She was pivotal in helping us navigate the intricacies of forming this collective from scratch, and doing so at an accelerated rate, given the urgency of the situation. We cannot thank this individual enough and are forever grateful for the instrumental role she played in improving our ImmunoX community. Please join us in congratulating Dr. Brianna Allen.
I'm not gonna cry. Thank you all so much. Thank you, Oscar. Thank you, the entire ImmunoDiverse team. I cannot express how much honor I have at having participated in the foundation of this group. And I'm so awed still daily by the dedication and the perseverance of all of our members. You've really stepped up to make your community better, make science better, and make this a space where everyone can come to the table and help support the change in this community, not just the underrepresented minorities who are struggling every day, but also all of the allies that want to step up and help make that change. And I know how labor intensive it is and how emotionally draining it can be to, dive into these conversations and really be leaders in this space. So cheers to all of you. And I can't wait to see how the group grows. And while I may have graduated, I still will check back in every once in a while. And I hope I can continue to be a mentor and support to the group and everyone who passes through it. So thanks again. And thank you to everyone who came to the first colloquia of ImmunoDiverse. I'm incredibly excited that this came together so beautifully.
Thank you very much, Dr. Allen. And now over to you, Dr. Cortez.
Hello, this is Victor Cortez of the Loxley Lab again. So I'll do the introduction for Dr. Herbert. Dr. Herbert, as he begins to get his slides ready. So it's my pleasure to introduce Dr. Dabrowski Herbert. Dr. Herbert has been an NIH-funded investigator since 2007. His laboratory explores the cellular and molecular networks controlling inflammation at the mucosal interface. His research program is particularly focused on parasitic elements, which are a major cause of morbidity in humans and animals of veterinary importance across the globe. Dr. Herbert has worked diligently on developing his leadership and mentorship skills during his tenure at multiple institutions, including the University of Cape Town, Cincinnati Children's, UCSF, and UPenn. He serves on scientific advisory and executive boards for the Keystone Symposia, IFI, and American Biological Society. His career has greatly benefited from a diverse assortment of mentors in the United States and abroad. Dr. Herbert feels it is not only his duty, but an honor to make time for those trainees who may need a little more nurturing, a little more compassion, and a little more candid advice during their formative years of scientific training. Today, Dr. Herbert will give a social keynote address to tell about his insights from his journey through science thus far. Please join me in welcoming Dr. Dabrowski Herbert.
Thank you very much, Victor. Put on my video and my mic. Can everyone hear me just fine? Yes?
Yeah, we can hear you.
Okay, great. Yeah, I am just so humbled by the invitation and just this colloquium today. You guys are just awesome in so many different ways. Yeah, I won't belabor that. I have a feeling of overwhelming emotion right now. I really do. I prepared a set of slides that this kind of goes through my walk up to today. I will say that this path has been a journey of becoming. I always feel like I'm just getting started. I don't feel like I've done my best science yet, but it's been amazing. And I hope it continues to be so. This first slide just, of the globe just really impresses upon everyone just how connected we all are. I'm not going to go into politics, but we now know how affected we are by things happening across the globe. We are in a very small space and we all share it. I had a really transformative part of my development when I had my genealogy done. And, growing up in South Mississippi, I just thought I was just descended from slaves. And while that is true, you can see here how diverse even my gene pool is. I had a little chuckle when I saw the Great Britain there, right? And I'm sure many of us share very complex genealogies that shape our individual identities. And it is my great hope that our individual identities become accepted and advertised just very frequently, and we don't have to shy away from them like I once did. So, about me, it's really can be summed up by this Robert Frost quote from one of his poems, in that I shall tell this with a sigh, somewhere in the ages and ages hence, two roads diverge in the wood, and I took the one less traveled, and that has made all the difference. My path has really been on this road less traveled, and I think it's led to a very fantastic experiences. I've not gone the traditional route. In graduate school, I started at Thomas Jefferson University of a stint of being out of school, where I was actually a blue collar worker for a while. That was a great motivation for me to solidify my intent on becoming an academic. David Abraham was my PhD supervisor, and David really was an amazing mentor. And I'm going to come back to this mentor issue He gave me a lot of freedom to explore ideas that he knew were not going to work, and he allowed me to challenge him to try to establish whether they're going to work or not. And then I finally decided it was time to finish grad school after trying a lot of rogue ideas. My broad interest for as long as I can remember have been focused on parasitic worms. And at that time, eosinophils and IgE, that's all we knew about type 2 immune responses. But of course, we heard the March DC talk a few moments ago, and the type 2 immunity has become very complex and intricate, and we now know it is not a default response. Actually, it's a deliberate response. And there are many very talented investigators across the field that are studying type 2 immunity. Our group has made certainly important contributions in this area, and if you're interested, certainly look me up for a postdoc. So I'm sitting in graduate school, and the senior grad student was a friend. We were playing chess. Maybe this is my second year, and he says to me, "Dabrowski, have you heard about this book, the bell curve?" Ha 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 ha. Now this was a friend, right? So I was used to, at that time, the racial jokes and being carded at frat parties that thought I didn't go to Jefferson, and my friends with the security guards. But, to have someone who was in my own lab, considered himself a friend, throw this in my face was pretty profound. I would say in 1995, 96, what was acceptable in academia, my thesis to you today is damn sure going to change, and it is changing. And you would know that this is pseudoscience and very controversial. And I said, well, look at the data. And the data, what does it say? I mean, from as far back as the turn of the 19th century, endless contributions of people look just like me, making contributions to society. And I want to point out Kizzmekia Corbett right now, who's really spearheaded, Anthony Fauci, one of our leaders in our discipline, pointed her out as really leading the COVID vaccine development. And, this aversion of people of color to trust the medical community rather, for developments because of past atrocities needs to stop. Because we are here and we are making amazing contributions, and not just contributions, but we're in the front. And so I really want to lift up Kizzmekia today, and you guys should definitely have her in the next colloquia. I'm sure that she has a story to tell. So I left grad school pretty fed up with American culture, I might add. Leaving the South and coming to the East Coast was quite a cultural shock, but finished my degree, and I said, well, I'm going to Africa because it's all better there. I'm going home, right? And it just so happened that in 1994, after South Africa had become a democratic country by many individuals' efforts, but mostly here highlighting Madiba, as he's locally known, of Nelson Mandela, that really opened up this country and made it possible for someone like me to go and do basic science research. And that's exactly what I did. And I must say that it was an amazing road trip. I mean, I know now what it's like to work with P32 without a Geiger counter. I'll just put it to you like that. Okay. But out of that experience, we made a very important insight, and one that I hadn't realized how impactful it would be on my career until many, many years later. And, this M1, M2 paradigm that is kind of passé by now was a theory at the point that we published this work. And it has continued to be cited. And I think one of the challenges of people of color is that citation index, and so I will encourage those of you, the faculty and the senior people who are still paying attention. It is really that citation index that's so important to recognize your colleagues. And so, I have a very special place in my heart for this institute here, the Marine Biological Laboratory. It was the first place where I attended a scientific conference. I've gone back many times. And it was pioneered by, there were studies pioneered by this scientist here, Ernest E. Just. You should know him. At the turn of the 20th century, he was doing phenomenal work in the area of fertilization and marine biology, parthenogenesis. And I always, when my lowest points, I must always go back to Just and ask, well, what must his experience been like compared to what my experiences have been like? And I won't read the quote here, but you can quickly just get a sense for someone who experienced so much discrimination just to do what he felt compelled and passionate about. He still had this love of humanity and of nature. And so after that time, I went on to Ohio and I started to build my program. I actually, let me go back for a minute. I went to the MBL and I submitted 2 abstracts. And the meeting organizer at that time, kind of in passing said, what do you get off giving 2 abstracts? I mean, 2 abstracts. Well, they were both accepted, And there was also a European individual who also gave 2 talks. So I went back to the organizer at the end of the meeting. I said, well, did you give this person such a hard time as you gave me? And he said, well, that person wasn't Black, and just walked away. That was what was acceptable at that time. It is no longer acceptable today because groups like yours are changing the paradigm. So again, I salute you. So going to Ohio, and now Fred Finkelman, really a king, really one of the leaders in the field of type 2 immunity, helped to solidify the importance of IL-4 and type 2 immune responses. And Fred really taught me about being passionate about the work, really strong advocacy. And I learned a lot from Fred. And I'm forever grateful for his mentorship. And I still today recently received a promotion. I still, reach out to Fred at this day. And that's what mentorship about. You never really stop being that mentor. It's like a parent. You never really stop, right? And so Fred and I did some pretty amazing work, all centered on parasites. We focused on brown beta and so on and so forth. But it was time for me to grow up, right? And so I struck out on my own. I started to build my laboratory while still trying to figure out, well, what is my identity? My identity, not only as an African-American male, but as a research scientist. So I dug deep and talked to a lot of people. I encourage you to constantly talk to your peers and your mentors and people outside of science. So here's my first group. You can see that we're all so happy to be wrapped in this worm. And so I developed a few paradigms to build this team. We were functioning as a unit. Strategically collaborate. Not collaborate with everyone, but make sure it's impactful. You're going to be recognized for that work and that you are respected for your ideas. Establish your own niche. The Burroughs Welcome has a lot of literature on this. Establish your own unique niche so you can get outside of the shadow of very prominent investigators like Fred Finkelman certainly was. And of course, yeah, if you can, get yourself a lab mascot. Not everybody can get a large worm like we can. I'm a little lucky there. And another nugget I want to leave you with is to build your road. Know what your particular talents are. Are you the front driver? Are you wanting to go into an area where there's really no one? Do you want to maybe go into an area that is somewhat well studied, but you feel like you have some important contributions? Or are you the type of scientist that's all about the really fine details and solidifying a paradigm? I just so happen to be a front loader, and I've accepted that. It's drawn criticism, but I love going into completely new areas. And as and that one of the reasons why I do that, because I love worms so much, and I started to develop this thesis that I wanted to drive our experiment. So mammalian coevolutionary adaptation to helminths has selected tissue repair to become immunoregulatory. Be bold and state your ideas, put them up there. No, but you don't need validation from anyone. And I didn't ask for validation when I started putting this up front and center as I was making tenure in Cincinnati, or when I got on the tenure track and started to give my talks. But it was really when I sat in an immunology it was much like an immunology retreat. And one of the investigators, got up and said, well, there's this molecule, trefoil factors, it's involved in tissue repair. We think is important for allergic responses, but we don't have a mechanism. And I knew at that moment that this is what I really wanted to start to focus my lab on. Nothing was known about the immunology of trefoil factors. Even to this day, we're struggling to build the tools to really do the fine-tuned molecular mechanistic work that, much like the talks you heard a little bit earlier today, I still remain envious. So we started to work on this repair factor and dig into the immunology. So could hookworm infections provide insights to tissue repair as mediated by trefoil factors? And indeed, it was quite fruitful, I might say. And so Marcia and I, who happened to be my chair at that time, and I want to take a moment to really lift up Marcia. Marcia Wills-Karp really fought hard to be recognized in the area of immunology and allergic diseases, and she is quite a prominent investigator in her own right. Marcia at that time was my chair, but you will notice the author list. We worked on this together. Marcia was completely okay with me being last author at a time in my career where most senior investigators would say, what are you talking about? You're just starting your lab. You're not going to be first author. I'm the chair. I'm going to be last author. This is a mark of someone who's really committed to your career and building you. Recognize it. Push for it. You can get it. And so with culmination of that work and others led to, model building, which is so important for biomedical research, right? Build your model, try to break that model. What's left is truth. I learned that from Ike Eisenlohr during graduate school. And so we started to build this idea that either worms or allergens disrupt the mucosal barrier, and trefoil factors, at least here, trefoil factor 2 was the star of the show, lead to the production of IL-33 from myeloid cells. And that was quite controversial at the time, but I'm going to come back to that in a minute, and just set up this whole loop of how type 2 immunity, one of the mechanisms of type 2 immunity at the mucosal barrier. So we worked on that for quite some time, and it was very fruitful in Cincinnati, but at the time that it was time to go, because of course, Fred was still there. So there was some issue about how independent am I going to be, recognized for still being in the place from where I trained. So UCSF came calling, but before UCSF calling, and we started to get recognized for this work. So I get invited as one of the speakers for a Keystone Symposium meeting. And I am on the board of directors. If you're interested in getting more ties with Keystone, please contact me. Off to Canada, to Banff we go, right? And I'm just so excited because, I'm still early days, but I'm a speaker at Keystone Symposia. And it was outside of this hotel that I had my whole kit on for snowboarding. It was through science that I learned how to snowboard, for I grew up in South Mississippi. And while waiting for the bus, I was with a couple of colleagues with my iPod in my ear, and this bus driver came up to me and dropped 2 suitcases at my feet and said, "What are you standing around for? Get these bags." The conversation stopped. I was with 5 other of my colleagues. They all knew I was an invited speaker. I was a cool dude, right? All of a sudden, I was reverted back to servant class just by the look of my skin. Now, those colleagues didn't say anything. They just kind of looked like, oh, wow, sucks to be you. That is got to change. Allyship, advocacy that we're talking about today, that's got to change. That shouldn't happen never again. For it did throw me off for a while, but after I gave the talk and those applause came, it kind of waned. Then I went to UCSF. And at UCSF, that it's a top tier institution. You can see the caliber of investigators that we heard from today. It's the competition is fierce. So I asked myself, am I going to go the easy road again, or am I going to take on the toughest question in my field? And the toughest question was, what was the trefoil factor receptor? They had been looking for it for 40 years. Who was I to think that we could identify a trefoil receptor? But I am Dabrowski, as you are you, and we identified one. We took a very radical approach. My chair at the time looked at me and said, "What? You're not doing real science." And we identified a trefoil receptor. So much so that I went on to the Burroughs Wellcome to compete with this idea, this radical idea that we could change the face of this field. Yes, I can. And I came back after that competition all really full of joy. And if you can see here that the molecule we identified was LINGO-2. And I practiced with my chair. You know, we had a very small division at the time. And he says, "So how did it go?" I said, "They love the LINGO project." He said, "What?" I said, "The LINGO project." I practiced. I said, "Oh, I thought you were rapping." "Oh, now I'm a rapper. You recruited me, but I'm a rapper." I did not let that one pass. I must say, I had grown in my skin and did not let that one pass. I'll leave it at that. So from UCSF, I traveled back east, kind of coming home, I would say, and I find myself at Penn. And I must say, I have never felt as comfortable and in my own right as I do here at Penn. The students are absolutely amazing. The UCSF students are almost amazing. and just so I say that the two of these individuals here are my grad students. Bonnie believes very seriously in advocacy, so much so that in outreach, so much so she's not going to do a conventional postdoc. She's going to go right into outreach. And that is the type of passion and fervor that we need. And it's happening. It is happening. So here at Penn, we're really diving into this IL-33 story. And again, here's the paradigm. As late as only about 5 years ago, that IL-33 is released from dead and dying cells, and then it acts on myeloid cells, lymphocytes, ILC2s to exert its biological activity. Was this really correct? Was it really correct? Because in 2012, we actually found that 33 came from myeloid cells. So be bold in your science. Yes, we're going to go after this. We're going to try to break this. So we form these questions. Are there other important sources of 33? Is there a specific mechanism? Everybody was looking at, we were in Belgium before the pandemic, and there was 400 people in this type 2 immunity room. Nobody knows how IL-33 release is mediated. We found it. We found something very, very interesting. Brian and I were talking about Preferentially a couple of weeks ago. And you know, that's what I found is my identity. Go where there's nobody else. Change the paradigm. Don't just patch up a little bit. Don't just add to existing picture. Build your own picture. Have the field follow you. And that's exactly what needs to happen now. That we need to revise this IL-33 model as just being a damage release molecule that promotes inflammation and ILC2 expansion and the like. But actually, there's nature's quite elegant, right? In that there's another pathway where myeloid cells produce IL-33, and this promotes Treg expansion, particularly the ST2 positive Treg subset. And so here you have to now appreciate how a single cytokine can not only promote inflammation, but can also be immunosuppressive. And so it is changing. And that is really my message to you today, ImmunoDiverse, that it is changing. You know, individuals like this, even though they're kings in our field, are no longer being able to get away with the crap that comes out of their mouths. It is changing. You've got to read about Ben Barnes. And if you haven't, it is such a phenomenal read. Barbara DeBen, and such a fervent advocate for women in science and just for diversity in general. This is what we need, right? We need that allyship, that advocacy for people who look like those who previously would discriminate against us, now to flipping the script and having a whole new different message. NIH is going to change. Foundations are going to change. What's acceptable to say to one another in the academic environment and to your students, it's changing. Because we now appreciate that not only is there diversity in the immune system, and they all work together in this really coordinated fashion, but there's important diversity in science, in our field. We all have important contributions to make. No one is bigger or smaller than other. We are a community, and it is together that we change the face of science. And so my group is quite colorful, and you know, it's so surprising to me how everyone lets me wrap them in this large stuffed worm. They all seem to be so happy. Well, not my first grad student, Kelly. She never really liked to be wrapped in a worm. You could see from her face, but we have a good time. And that is my style. I think, you know, doing phenomenal science is really great. But if you're under undue stress doing it, what's the point? You got to have some time to regenerate, to replenish those stores. And that's what we do in my laboratory. So let me just leave you with this, and we can wrap up for the Q&A. I wrote this for Black and Immuno, and I kind of tweaked it a little bit for today. And I'm going to recite it for you. To my sharpest minds and those considered not so bright, to my people of the lightest hue and those that can pass for night, let your light shine and do not apologize for being uniquely you. Assimilate only in excellence, but in nothing else that you do. And when they try to make it seem that you are a source of sympathy, know that the Black tax has built your stamina to reach infinity. When there seems not enough known or no reagents to test that condition, know that the only block to progress is your willingness and your volition. Since the middle passage, thriving against all odds, is our specialty, use those passage atrocities as brick and mortar to shield against mental instability. So stand tall, ImmunoDiverse, speak confident and clear, pipette with precision. Humanity awaits your contributions of talent and perspective. Just make your decision. Thank you very much.
Okay, sorry about that wait. I thought Victor might be coming on, but I can go ahead and take it. So thank you so much, Dr. Herbert. I heard that passage that you read at Black and Immuno, and hearing it again now, it's just as impactful. So thank you for those words and the inspiration, and for sharing your story. I know I learned a lot from it. We are taking questions for Dr. Herbert, so please send them in the chat to Question Master, but I'll go ahead and get us started. So the first question is, would you do anything differently in your journey if you had a redo?
Would I do anything different in my journey if I had to redo? No, I really don't. Every step in this path has been important for my development. It shaped me to be who I am today. I don't really believe in regrets. I really believe in learning from your mistakes, changing them, and doing it different in the future. Yeah.
Love that answer. Second question, actually a follow-up to that. What do you hope your legacy will be?
I hope my legacy is to give more than I took, to be shoulders that others can stand on, to be an inspiration for someone who, Byron, right? MIT and I mean, my path has been very, very different, but here I am, right? I think that's the legacy, with grit, confidence, personality, you could go pretty damn far.
Yeah, well, I would say that to me, you're kind of doing a lot for us for today, sharing with us in giving. So thank you again for that. Next question, are there special considerations when writing a grant on a paradigm shifting idea?
Are there special considerations? Yes. So, you know, it's notorious for those established in the field to reject new paradigms, right? New ideas. It's hard for us as a community to accept something is different. So the tact I've taken is to relate the new finding, the paradigm shift, to an established fact. You really need to say, yes, the established part, there is some truth to it, but let's stretch into this new direction. If so when you're writing a grant, you don't want to just come out, oh, well, say for instance, Perforin 2 is just the conduit and it just, it does all these fantastic, you need to build a story. Scientific storytelling is real and it takes skill to build it. It's a story, not just a, something out of the sky, right? So
right.
make sure you're putting it in context and then stretch the paradigm. That's at least for my experience, what's been important. The Trefoil grant was, it scored amazingly at the time, just because it was completely new. But IL-33 had started to garner a lot of interest. People were trying to understand how to regulate it. So we showed established roles for IL-33 eosinophils, and then Trefoils did something totally different. So,
right. Thanks. Next question. I'm sure you have touched on countless students' lives. Are there any occasions where you learned from your mentees?
All the time. All the time. Oh my gosh. Yeah. I mean, one of the things about my experience at UCSF was, I learned kind of my style. I didn't get a graduate student of my own, but I had lots of rotation students. There's a lot of reasons for that. My lab was in a weird location. But it was at Penn, we're having my first graduate student. Wow. I thought I knew what I was doing, but wow, Kelly taught me so much, So I'm constantly learning my own patience and tolerance and level of insight, where I reach my limits, So yes, it is absolutely 360.
I have a follow-up question for that as someone who hopes to one day be a PI. Are there any tricks for ensuring that you're always able to learn from your mentees? Like, how do you stay in tune with them and, really listening, if that makes sense? I guess maybe it just comes naturally.
Yeah, it's yes and no, right? Because yes and no, because we all have different tolerances, and the mentor-mentee relationship is a lot about chemistry, right? When you rotate through someone's lab, it's just as much about the science, or it should be just as much about the science as it is the connection, feeling heard, right? And so, having the student who really speaks up for themselves and say, hey, I'm not feeling heard here, or, what you're saying to me is just not sitting right, then as a mentor, you get to ask yourself, okay, how am I going to deal with it? Am I going to say, oh, well, come on, either you're, bend to my will and come along to my way of thinking, or you get out of my lab? Or is it, maybe how can I be more effective here? Once you recognize that there's some disconnect, because some relationships, in science and otherwise, are just very fluid. Some you have to all of you have to work on, but some of you work on much more. And then some of them are just a no-go. And so being able to very quickly recognize that that is I think that's an acquired skill over time, right? Recognizing where you are, how far can you go? You need to know your limits. A lot of knowing yourself is important. So that's why I'm saying a coming to being is what this path has been like for me. So, and like I said, I'm having the best time in my life right now.
Awesome. Well, our next question actually comes from one of our trainee speakers, Carlos. He says, amazing, Dr. Herbert, you have truly contributed to the type 2 immunity field, and you should feel very proud of your contribution. Do you think that the PI mentorship style matters for URM and peer students to succeed in sciences?
I want to make sure I understand the question. Do I think that the PI mentorship style can you just repeat it? I really want to know what.
Yeah, I'll repeat it as is, and then we can talk through it if that helps. Do you think that the PI mentorship style matters in order for URM and peer students to succeed in sciences? I guess, are there some PI mentorship styles that will work better than others for peer students to succeed?
Yeah, I mean, I really want to avoid putting URM scientists as a singularity, right?
Yeah.
Because how we may look some certain way, our backgrounds could be very, very different and everything, right? So everyone is unique. I think just active listening is so important. I'm dealing with a situation now. There's just happens to be a student, because I do a lot of mentoring where it doesn't directly benefit me. Mentor students who are in other labs, and they just kind of reach out to me because they know I've been, as a visible, Black man, doing science. I know I've gone through a lot of experiences, and so this one student is feeling not heard by the mentor. It just happens to be, kind of the mentor is Caucasian and the student is Black, and there may be some situation there where a different approach to speaking with the student. I mean, if it continues on, I think I'll probably pull the PI aside and say, hey, this student is really not feeling the chemistry of the lab, and kind of maybe there's something that you can do, because not a one size fits all just doesn't work. I think that's really how I want to answer the question. It's not about something unique to the URM scientists. It's just, I don't think in mentor-mentee relationships, one size fits all, right? Because that's the whole point of this thing, right? We're diverse. Like someone who is, morbidly obese doing research is just going to have a different perspective, going to have a different set of needs than someone who's, not. So I don't want to go into the, okay, for URM science, you need to do this for them. They're like special and different. I don't ascribe to that personally. Somebody else may have a very different opinion. I think it's in each and every situation. So adaptability, right? Being able to recognize a person's needs, and maybe in the relationship how it needs some tuning on both sides is really important.
Great, awesome. Well, that's unfortunately all the time that we have for questions for you today. Dr. Herbert, I just want to thank you again so much for returning virtually to UCSF for a second time this year to tell us more about your story. We are all really lucky and grateful for the time you've taken to share your thoughts and inspiration.
It is my honor to be here.
Thank you.
And I'm happy to continue this discussion one-on-one for anyone who feels like right? Because I was that student that needed a little bit more mentoring, a little bit more candid advice. So that's why, that's why my commitment today. So thank you all.
Thank you. Before we end this event, we would like to close with some breakout discussion rooms. The goal of these conversations is to learn and to normalize discussions about systemic racism in our research institutions. and to develop knowledge and initiatives to combat it. Your discussion moderator will have some questions to get the conversation started. Please be respectful of each other and mindful of people's different perspectives and identities. We encourage you all at this time to turn on your video if you feel comfortable and join your assigned rooms. See you all back here at 5:13.
Hey, John, did you want to participate in this? Just a couple of questions.
I'm good. Thank you, though.
Yeah, I kind of want to take a quick break too.
Yeah.
Oh, hold on.
We want to close by again thanking all of our training speakers, as well as Dr. Bryson and Dr. Herbert, for taking the time to share your science and experiences with us. And finally, thank you all for coming. Together, we make real change. Have a good weekend, everyone. Good night.